टीम को पुलिस की कर रहा है ऐसे क्लोज कर
Sir, my voice is audible. Hello.
यस मैम यू आर ऑडिबल
Okay.
इसमें आज 11 तारीख है ना, डे 3। इसमें आप जाओ IRS 2024। हाँ, इसमें यहाँ भारतीय तिरंगा। इमोशनल नहीं छोड़ के गए, ये तो चलता भी नहीं है।
चलता है
करो, नेक्स्ट करना स्लाइड में, नहीं हो रहा है। पॉइंटर है ये। और माइक्रोफोन भी नहीं था, वो कॉलर माइक यहाँ पर।
अभी कुछ आपको टेस्ट करना है तो ठीक है, लेकिन हो रहा है।
अरे कल नंबर था ना, कॉलर माइक नहीं था ना कल।
नहीं था, नहीं था, ऐसे चलेगा क्या करना है? अच्छा हाँ, शाम को नहीं रख सकते ना, ये चार्जिंग पे चलेगा। ये चला
अच्छा
ये पूरे दिन चलता है। ठीक से ये कौन सी था नीचे आओ। सर्विस आया तो आगे आवाज रिकॉर्ड की कैमरे हो सकते हैं ना।
इसमें आपका ऑडियो लगा
सेकंड सेशन ठीक है बात। डे 3
ठीक है, छोड़ो कब लगा
पहले तो लगानी है 7th
मतलब सेशन एग्जैक्ट
नहीं
हाँ इसी में ओके।
फोटो
और
क्या करें, खटखट आएगी फिर लगाओ एक मिनट
इसे चला मैं
अब भी होगा थोड़ी क्या घटा रखा है।
दबा नहीं जी ये वाला
मत करो अभी तो कनेक्ट
ठीक थैंक यू।
सौरभ में IARS और
Obviously.
ऑब्वियसली
IARS
बराबर वीकनेस और ऑब्वियसली आएंगे
वो तो आएंगे ही।
ये लगा देंगे।
कहाँ लगेगा तो कैसे लगेगा?
फर्स्ट
मेरा अभी फर्स्ट नहीं है जो ऑनलाइन है
इनके
अच्छा, ऑन
पहले
करना
हम
पड़ेगा।
लोग के
यहाँ लगा
पहले
देंगे शर्ट पे।
और बस
या
बस
माइक में
ये
बोल
हम
देंगे
दोनों
ज़्यादा
एक साथ हैं और ये पहले।
यार ऐसे बोले ना यहाँ पे
सौरभ का
मूवमेंट
फर्स्ट है।
मूवमेंट निकालना ना इसमें बोल देना इधर उधर मूवमेंट को
आप
निकालना
इसको
ना।
नहीं
अंडरस्कोर
नहीं,
वन
मैं
कर
को
दो
यहीं
तो
पे
ऊपर
बोल
चला
रहा
अभी
जाएगा।
यही यही
हूँ,
या
से
वन
पर वो
सौरभ
अगर
रहेगा।
सोच रहे हैं तो फिर अगर जैसे थोड़ा
तो
घूम
फिर कॉलम आई चाहिए ना।
हाँ तो
हाँ
इनको
मतलब
दे दीजिए, हम
हाँ
लोग
हाँ
नहीं इसी को
हमारे
दे
वो
दीजिए।
देख लेना। is session is on genetics and physiology. I invite my colleagues, Dr. Sandeep Gupta, to chair the session. I invite Dr. Landan Puri, Bhavani, to co-chair the session, and I invite Dr. Bhim Singh to act as a facilitator in this session. And we have 8 presentations. We have how many labs? So each lab has one presentation. No, 4 labs. So I presume that most of the presentations from the lab is going to air estimating areas. So over to you, Dr. Sandeep.
Thank you and good morning. So today is our first session. This is our genetic physiology. As was already informed that we have 3 presentations here, 5 people presenter. First one is from the background bar, Bhavani Tabar. So please online, Bhavani. Bhavani, are you there?
Yes, sir.
Bhavani Sagar has done her Master's in Geology from Dehradun University, Odisha. Currently, she is a PhD scholar in Tehran, Coimbatore. She is working on taxonomic characterization of selected Galliformes of India using feather morphomatics and DNA for application to wildlife sciences. Over to Bhavani.
Thank you, sir. Good morning, everyone present over here. Myself, Bhavani Sabar, working as a Junior Research Fellow and a PhD scholar, will be presenting on the topic titled Insights of Feather Microscopic Characteristics and Genetics of Select Galliform for Application in Wildlife Pharmacy. Galliforms are commonly known as game birds or gallinaceous birds, are terrestrial species which are renowned for their vibrant plumage, attractive adornment, and elusive behavior. Today, I will be sharing with you all some insights of the galliform feather in detail. Based on the 2011 IUCN Red List, out of 290 species, 76 species, that is 26%, face some serious threat due to hunting, trading, poaching, and illegal poisoning. Categorizing these facts, we set 3 objectives in an ongoing project to help the law enforcement agency to deal with the seizure cases. And these are as follows: feather microscopic database, DNA database, and to understand the ethnogeological practices of select galliform. In any case of galliform hunting, we found feather as a piece of evidence. So, however, essential reference database is not present for exact identification of that particular species. Therefore, our first aim is to develop a comprehensive database on the feather microscopic structure of select galliform. During fieldwork, we collected naturally shed feather and discarded non-viable egg sample from Padmaja Naidu Himalayan Zoological Park, Darjeeling, and Bannerghatta Biological Park, Bangalore. A total of 7 Indian galliform, along with 3 exotic species which are native to Southeast Asia and Europe, were collected. We followed a structured methodology. After collection of the feather, we segregated the feather sample into different types, and we further examined the feather in 2 different microscopes, that is light microscope and scanning electron microscope. We segregated the feather type into various feather types such as wing tail, that is flight feather, contour, and semi-plume. The Indian peafowl, commonly known as the national bird of India, is mostly used for the tail feather is mostly used for crafting prayer fan and also to craft the dream catchers. Offline markets during the festival are the main sources where we can get this tail modified contour feather of this Indian peafowl. The spectacular lady-amest pheasant is an exotic species but is mostly commonly found in most private aviaries in India. And because of this vibrant plumage, it is also kept as an ornamental pet for wealthy people. And their tail long heckle feather is mainly used for designing costume at carnivals. Not only in the offline market, but also in some e-commercial websites such as Amazon, Flipkart, and eBay, most of the body contour feathers are being sold, such as in this silver pheasant. Golden plumage of this golden pheasant is mostly used for fishing tires and is also used in the fashion industry to design various costumes. To study the structural basic of feather, we aim to measure the macroscopic and microscopic detail of select galliform. Diving into the microscopic detail, we observed hooklet pattern in the pinnaceous barbule, node and internode, bearing node and internode in the plumulaceous barbule. Imaging was conducted in both microscope, that is compound light microscope and field emission scanning electron microscope. For compound microscope, we use Labomade LX400 with a magnification level up to 400x, and field emission microscope, we prefer Jess and Jewel model for a magnification up to 9.70kx for feather examination. For microscopic preparation for light microscope, the microslide will prepared using two methods, that is dry and wet. For dry method, we used the nail varnish, and for wet method, we used DPX. For FESEM, different part of the feather like pinnaceous barb, plumulaceous barb, after feather, and cross section of the rachis were underwent into gold sputter coating and were placed in the aluminium stub, and carbon tape was attached for further examination. The result from the light microscopic resulted for like different color variation, as we can see in this Indian peafowl data. Blue-green iridescence were mostly found in the feather type of contour semi-plume, and no pattern was observed in the semi-plume and down. Also, for the lady amherst pheasant, different plumage of color was seen in the different feather type, and pattern was mostly observed in the contour and flight. Also, the morphometric measurements of lady amherst like calamus, rachis, vein, shaft length, and total length of the feather were taken. Silver pheasant feather resulted various pattern in their contour feathers because they are having more black and white stripe in their whole plumage. The golden pheasant revealed orangish and brown color in their plumage, and no pattern was observed in the contour, semi-plume, and flight. And two barb type, that is pinnaceous and plumulaceous barb type, were taken for noting the presence absence of certain microscopic structure in the light microscope. Further, feathers were examined under various magnification, 40x, 100x, and 400 magnification under light microscope, and the following results were obtained. For the Indian peafowl, we saw structures such as subpinnaceous region, prominent nuclei, different type of ventral teeth, such as leaf-like and teeth-like, a prominent hooklet present in the contour and pinnaceous region. In the plumulaceous region, we found pigmented ring node and spine node. For the microstructure of lady amherst feather, we found ramus pigmentation in the proximal area of the pinnaceous barbule. Prominent nuclei with hooklets and ventral teeth were observed. When we use the wet mouth method, we clearly observe the colored pigment of the barbules in the pinnaceous region. And like large density of ring nodes were found in the lady amherst plumulaceous barbule, which resulted in multiple annuli known as the multiple ring nodes. For microstructure of silver pheasant, we saw transitional barbule, like we saw transparent barbules with pigmented barbules in the pinnaceous region. As in the third slide, we can see a total microstructure of a distal barbule showing the leaf-like ventral teeth, following by it is the 5 hooklets, and above to it is the ventral and dorsal cilia. And multiple ring node were also observed in the silver pheasant. Moving towards the golden pheasant, here we saw orangish barbule in the pinnaceous region along with the hooklet and ventral teeth, and very less amount of multiple ring nodes were observed in this. Comparing it to the light microscopic structure with the FESM micrograph, here we are confined to more about the hooklet pattern, the cross section of the rachis, and the different type of ring node and spine node observed in the plumulaceous region. So for Indian peafowl, long hooklets were observed in the pennaceous part, and multiple annuli, like multiple ring nodes, were prominently visible in the plumulaceous region along with regular spine node. For red jungle fowl, a structure known as pronged hemuli, which is not observed clearly in the light microscope, can be clearly observed in the FSR micrograph because of its high magnified structure. Uh, Also, in the red jungle fowl, spine nodes were more irregular as compared to the other galliforms. For Kalich Feason, we found that regular spine nodes were observed in the plumulaceous and also ring nodes were found. In the grey peacock Feason, we can see over a hooklet how they are like hooking to each other to form the greater barbule density in the pennaceous form. And also more amount of melanosomes were found in the cortex region of the rachis. The Himalayan monal showed more amount of melanosome compared to other galliform because of its pigmented color, feather, plumage, and the ring nodes were like greatly not evolved in the Himalayan monal, but in Temminck's tragopan, we can look over for the pronged hemuli as well as the irregular spine nodes with more longitudinal grooves in the internode. In grey jungle fowl, more amount of hooklets were observed, and in the lady MS, also pronged hemuli along with more expanded spine nodes were clearly visible. In silver Feason feathers, pronged hemuli was also observed. We found a multiple ring node of 7 to 8 in the plumulaceous region of the silver Feason feather. Here we can look over the distal and proximal arrangement of pennaceous portion in the silver Feason feather. And golden Feason also revealed a very neat, regular manner of spine node in the plumulaceous region. That was all about the feather microscopic database. Our second aim is to develop a DNA database that will help law enforcement agency for species identification. So Kaizen DNA extraction kit along with phenol chloroform isoamide method, both was used for extracting DNA from the collected egg sample. Also, the various egg morphometric measurements were taken, such as color, length, width, and weight of the egg before extracting the DNA. We took eggshell membrane, yolk, feather barb, rachis, calamus to extract the good quality of crude DNA. And opportunistically, we got developing chick in some of the egg, which added extra benefit to the high amount of quality of DNA. So these are the results of the egg morphometric, which reveal like Temminck's tragopan and Himalayan monal showed more egg pattern, like light spot and dark spot were clearly observed. And the egg weight of Himalayan monal was the highest as compared to the golden pheasant. Slight modification of the standard DNA extraction protocol method, that is Kaizen and phenylchloroform method, were used to quantify, were used to obtain the crude DNA, genomic DNA, and was further amplified into 3 mitochondrial genes, that is 12S rRNA, cytochrome B, and 16S rRNA. Further sequencing is still in process as we recently got blood sample for other Galliform species. Many indigenous tribes in India hunt galliform for their proteinaceous bushmeat, for many traditional practices, and are unaware of about the Wildlife Protection Act. Lastly, we aim to understand the ethnozoological practices related to galliforms in India. For that, a semi-structured questionnaire based on participatory rural appraisal method was conducted at Koraput district of Odisha and the forest area adjoining the Darjeeling city. A total of 45 responded interviewed in Odisha and 20 responded interviewed in Darjeeling. And we analyzed the result by using fidelity score. So fidelity score value refers from 1% to 100%, and like it is specifically used to quantify the use of specific animal for a particular use. So out of 13 galliform species, 8 galliform species were found in the Koraput as per the interview, which is used to treat 15 human ailments. Oral mode of application, that is 52.1%, was higher as compared to the topical mode of application. Looking for the fidelity level percentage, Feathers of red jungle fowl and domestic chicken was used mainly for ear ailment, which scored a fidelity score of 100%. And the three type of quail species, which is used to treat kidney stone, scored a FL value of 87.6%. Also, we documented various hunting trap for Galliform, including the net capturing method, catapulting method, and the bamboo basket trap method. To conclude the feather characteristic, both the microscopy, that is light microscopy and FESM microscopy, revealed various microstructures such as hooklet, cilia, and nucleus, ventral teeth, and various type of node. According to Dove and Koch, ring-shaped structure are distinctive characteristic of Galliform, and we found the ring-shaped node in all of the Galliform we study. In FESM, we focus mainly on the pronged hemuli and the cortex medulla and melanosome respectively. For the DNA database, amplification of crude DNA has been done for these samples from the collected egg sample using the 3 mitochondrial gene, and the DNA sequencing for other species are still in process. For ethnomedicinal practices, 8 species of Galliform were used in the Koraput district of Odisha, and 3 species of Galliform were used in the Darjeeling district. I would like to thank all of the following people and organizations who helped me in all possible way. Thank you.
So you can try the listen of Vani. So we'll call later on when we start to listen.
Sorry, sir, I couldn't hear.
Vani, you can wait till the next presentation is over. Then question will be right for you.
Sir, actually, the voice is not coming clear because of the mic.
You wait for some time, okay?
Okay,
sir. You wait till the second end for your question. Now, next question.
Finally, we can log off your video and screen, so you can wait for some time till the other two speakers finish, and then we'll call you for questions. Thank you, Bhavani. Next, we have Sora Kuhela, who will be presenting on the preliminary hormonal assessment of translocated cheetah population in Dugong National Park, Madhya Pradesh. And I request Dr. B to kindly introduce our speaker.
Saurabh holds a master degree in life sciences from Central Institute of Gujarat. After that, he joined DNHS as a biologist and worked on vulture conservation. Currently, he is working in Cheetah project as a project associate from October 2023. Over to Saurabh.
Hello? Hello?
Meanwhile,
Hello?
continue your question to the guy.
Thank you for the introduction. Good morning, everyone. Thank you for being here today. When we talk about big cats in Indian perspective, we generally came across these 4 species, that is tiger, lion, leopard, and snow leopard. But in the historic times, the big cats in India also included the cheetah, that is the fastest land mammal and an icon of nature. Historically, 5 subspecies of cheetahs were present in approximately 44 countries throughout Africa and Asia. But due to anthropogenic pressure, today the free-ranging cheetah population were only constricted to 10% of their former range. So, with a population of approximately 7,000 individuals remain in the wild globally. So here in the map you can see the lighter part showing the historic range of cheetah, and the darker part show the present range of cheetah. In India, historically, the cheetahs were present everywhere except the higher mountains, the coast, and the northeast region. But due to several factors such as coursing, bounty, sport hunting, poaching, habitat loss, and decline in prey base, the cheetah population in India were drastically declined. And the last cheetah was recorded in wild in India in the year of 1948. And in year 1952, the cheetahs were officially declared as extinct in India. So after 70 years, Government of India took the initiative to bring back cheetah from Africa to India and translocated them to Kuno National Park, Madhya Pradesh. So far, the translocation has been done in two phases. First, in September 2022, when three males and five females were arrived from Namibia, and in second phase, that was in February 2023, where 7 males and 5 females were arrived from South Africa. The idea behind the translocation to establish viable cheetah population in India. The Project Cheetah is the first intercontinental reintroduction of a wild large carnivore species. So it is not surprising that the project of such complexity would face many challenges as the animal need to adapt to the newer environment. To counteract all the challenges and in order to achieve the goal, out of several important factors such as habitat, prey, co-predator, behavior, ranging pattern and health, studying physiological parameters is also an essential component. Several studies incorporated physiological assessment of animals after their successful translocation to new habitat to assess the possible physiological change and also help in conservation process. Hence, my study deals with preliminary hormonal assessment of translocated cheetah population. Literature suggests the use of 3 parameters, that is stress, nutrition, and reproductive health, to monitor the wild population. Stress corresponds to the physiological stress in which high glucocorticoid or GC level indicates high stress and low GC level indicates low stress. In opposite to that, in case of triiodothyronine, that is T3, high T3 level indicates good nutrition in animal and low T3 indicates poor nutrition. In case of reproductive hormones, which indicates the reproductive status of animal, in case of testosterone, high testosterone indicates male reproductive phase, high progesterone indicates female reproductive phase, and high estradiol indicates female during estrus. Interestingly, all the 3 hormones released in both the sexes. To avoid further stress to the animal, we avoid invasive sampling such as blood and took a non-invasive sampling approach and used fecal samples of cheetah to measure the hormone metabolite. Measuring hormone using fecal samples is not an easy job and requires substantial processing as well as validation to generate the reliable data. So after collection of samples, the samples were thawed, pulverized, sieved, and the hormone metabolites were extracted from them. After that, hormone quantification was done using the ELISA assay. Location crate, and after the translocation they were shifted to the quarantine boma, which is a smaller enclosure, and after the successful completion of their quarantine period they were shifted to larger boma, which is a pretty large enclosure and have conditions similar to the wild. So our samples were collected from all 3 facilities and that provide us with a tremendous opportunity to assess the hormone level at all 3 environments. So we hypothesized that we will observe high GC level in crate as it was expected the animals were in higher stress during the translocation process. Our another hypothesis is that in case of T3, we will observe the low T3 level in the larger boma as with compared to the larger boma in other two categories. Cheetah also gave birth to cubs for the first time after translocation, and which gave us another chance to study the reproductive status. So before moving to the next slide, I just want to say it is a very proud moment for all of us that we are witnessing the successful breeding of cheetah and these cute cubs on our grounds after several years. So a total of 160 samples from all three facilities were collected using sterile Ziploc bags by the field team at Kuno, out of which 64 belongs to male and 96 belongs to female. So after successful collection, the samples were sent to Wildlife Endocrinology facility at WI, where the samples were stored at minus 20 degrees Celsius before the further processing. So here are the results of biochemical validation of all 5 hormones, in which the working dilution for each hormone is different. And that's why the biochemical validation of biochemical validation of ELISA kits is very much essential before the hormone metabolite quantification. So in case of GC, the working dilution is 1:1280. For T3, the dilution is 1:40. For progesterone, the dilution is 1:200. For estradiol, the dilution is 1:80. And for testosterone, the dilution is 1:160. So as I said earlier, the cheetahs were housed in 3 facilities. We thus compare level of glucocorticoid and triiodothyronine of all the facilities. Further, we also segregated our analysis between male and female. So here we observe low GC level in crate as compared to the quarantine boma and larger boma, which is opposite to our hypothesis, as we expected animal to be high stress during translocation. This could be possible because during translocation, the animals were under the effect of long-acting tranquilizers, which suppress the stress and anxiety in animal. Here we observe low T3 level in larger boma, which supports our hypothesis, as in the larger boma, the conditions are very similar to wild, where prey is available, but animal need to hunt on their own, while in other facilities, the food was provided according to the protocol and biological need of the animal. Here we do not observe any GC level difference in male and female of crate sample. Meanwhile, we observe high GC level in females in both quarantine and larger boma, indicating high sensitivity of females to stress than males. Here, we do not observe any differences in the T3 level between the sexes in all 3 facilities. In case of reproductive hormones, for progesterone and estradiol, we did the individual-wise analysis among females. We also did the pair-wise analysis between males and females for all 3 reproductive hormones. So here for progesterone, we observed overall variation in the progesterone level of all females. However, the substantial variation was concentrated within a small subset of females, indicated by the red box. In case of progesterone, we also have a very interesting observation that by using an iterative process, we have created a baseline that separates the progesterone level of pregnant female and non-pregnant female. So here in our case, only 3 females show progesterone level above the baseline, that is Asha, Gamini and Jwala, and these are the only females who gave birth to cubs after translocation to Kuno National Park. Here for estradiol, we observed overall variation in the estradiol level of all females. However, here also the substantial variation was concentrated within a small subset of females, indicated by the red box. Here in the pairwise analysis of all 3 reproductive hormones between male and female, surprisingly, we do not observe any sex-specific differences in the reproductive hormones. So here the conclusion of this preliminary study is that we observed low stress level in crate as compared to the other 2 facilities, possibility due to the effect of long-acting tranquilizers. Result indicates that females are more sensitive to stress than males in both quarantine as well as larger boma. Low T3 level in larger puma might be due to the feeding practice in the other facilities. We observed no differences in the nutritional status between the sexes, and the progesterone measure in pregnant female could be used to ascertain their reproductive phase. So here is the way forward of our study, that more systematic sex-specific field sampling for reproductive hormones in the field would be very beneficial to categorize the animal into different reproductive stages. Long-term monitoring of hormone level, both spatial and temporal, will be critical for future management of the animals in the wild. And implementation of these approaches post-release in wild will help us in future to monitor the hormone levels of cheetah. So I would like to acknowledge all the members of the project for their help and support. Thank you.
So we will call the next speaker. That is, we have Bhati Aruna and Priyanka Prasthita. They will speak of the mystic tales of high altitude mammals that we find inside the water and that Panda at Dugong Zoo.
Dr. Bharti has completed her PhD in reproductive physiology of Chinese anatomy. She studied different endocrinological parameters in different sectors. When not working in labs, she loves to experiment with different cuisine and engage in outdoor sports. Over to Bharti.
A very good morning to all. So today I am thinking that from past 2 days, everybody is learning a lot about ecology. I know these past 2 days have been quite intensive. So I would say just take a little pause and detour to understand the physiology of some high altitude mammals. So today I take this opportunity to shed some light on the endocrinological correlates of markhor and red panda. So the segment of this presentation, or I can say the roadmap of this presentation, is divided into 2 segments. The first segment represents the reproductive biology of markhor, that will be presented by me, and the second segment represents the physiological stressors in red panda, that will be discussed by Triambak. So without further ado, let's get started. So what is markhor? Basically, markhor is a medium-sized mountain ungulate which is sexually dimorphic, and it is distributed across the mountains ranging from 600 meter to 36, am I audible? 3,600 meters in the Himalayas. It is widely categorized into 3 subspecies, and recently it entered the IUCN list of near threatened in 2015. So thanks to the multidisciplinary conservation efforts such as protected area management, community initiatives, limiting hunting and poaching, and conservation breeding, these approaches help the population to recover, which actually reflected in the IUCN list. However, the conservation breeding programs have mainly focused on assisted reproductive techniques such as artificial insemination. However, the success and the optimization of any breeding program depends on the multidisciplinary approach as well as a profound knowledge of reproductive endocrinology, because the reproductive cycles across different taxa is highly conserved. However, do we have any background information on markhor reproductive behavior? A little, yes, because the behavior has classified into different sections such as rut, non-rut, rejoining of males and females, gestation and parturition. Over here, it shows the chronology of different events that have been represented in the wild. So the males show rut during the month of November. The gestation is approximately of 155 days, maybe from November until March or late April, and the parturition is seen in the month of late April and early May. So this particular information gives us the opportunity to validate our endocrinological findings using non-invasive sampling. To initiate this project, to understand the reproductive biology of markhor using the reproductive hormones such as progesterone and testosterone, we collaborated with Darjeeling Zoo. So Darjeeling Zoo is the only highly specialized high altitude zoo in the country. Not only that, it's a national coordinating body for wildlife conservation breeding programs for 4 different species, namely red panda, Himalayan salamanders, snow leopard, and Tibetan wolves. For this study, we selected 5 individuals of Markhor, 3 females and 2 males, namely Ivy, Sonia, Supriya, and for males, Ranbir and Urban, of subspecies Capra falconeri heptaneri. Although for the study, the fecal sampling was highly sex specific. For males, it was 1 to 2 samples per individual per week, and for females, it was 2 to 3 samples per individual per week for a duration of 1 year from May 2022 to April 2023. So, in order to fulfill our goals of assessment of hormones, the study aimed to to standardize the reproductive hormones such as testosterone and progesterone using the fecal matrix, followed by the characterization of reproductive phase in both the sexes. The protocol that has been explained by my friend earlier is very similar for this study, but I will quickly walk you through the same. So the process was initiated by drying and sieving, followed by extraction of fecal hormone extracts, which was followed by standardization that involved 2 processes such as parallelism and accuracy. Once the standardization is done, we quantify the hormones using enzyme immunoassay, and once we have the data in our hand, we use the software or the package in R called hormonal. So the standardization of progesterone in females suggested that 1:240 dilution is the appropriate dilution in finding out the progesterone concentration in both the sexes, in males and females. Similarly, a working dilution of 1:120 was found out to be more suitable to identify the testosterone concentration in both sexes, male and female, using the fecal extracts. So once the standardization is done, our next step is to quantify the remaining fecal extracts. So the remaining fecal extracts were quantified using the process called enzyme immunoassay. So after quantification, our data was categorized into different months. After the categorization of data into different months, we used the package in R called hormonal that helped us to understand what is happening here. So hormonal is a package that basically overlays the hormone profiles by exploring the correlation between different phases. Let's take, for example, here, if a female is pregnant, then it identifies the correlation between pregnancy, gestation, parturition, etc. by building the baselines. Now you must be wondering, what is a baseline? So baseline, basically, it's a value that differentiates between the high concentration and the low concentration, basically the circulating hormones in the body. So after conducting the analysis, we found out the dotted line over here shows the baseline in the female, and it was very individual specific. That shows the values which are above the baseline represents the reproductive phase in the females, and the values which are below the baseline represents the non-reproductive phase. Interestingly, despite having all the baselines different in all the females, we still found out that the reproductive phase in all 3 females ranges from October through April, whereas the non-reproductive phase in males ranges from May to September. In addition to that, there was a significant difference between the progesterone level that actually helped us to classify the 2 phases. Similarly, for the males, the baseline was also very individual specific. Despite having individual variations, we could see the reproductive phase in males ranged from September to November, and the remaining months were classified as the non-reproductive months. So now, do you think that the data that we have collected complemented the data which is collected by the field biologist? I can say certainly yes. So in the earlier slide, I showed that the ecological reproductive behavior of this species looks like this. But after quantifying the hormones, we could see the reproductive phases or the different phases in the reproduction of male and female in captivity also shows an overlap with the observations in the wild. So let me give you an example. So the parturition in the wild was between late April and early May. A very similar observation was observed in captivity as well. and as well as the gestation phase in female ranged for 155 days in the wild. So for us, the gestation phase was around 200 days in captivity, whereas all the orange bars shows the non-reproductive phase, very similar non-reproductive phase in all three individuals. So now I'll pass on the mic to Trembak. Oh no, let me finish. So I can, for conclusion, I could say that the data which is collected in the the data collected in captivity holds a very equal significant importance as the sample collected in the wild, because it also helps us to fill the missing gaps which cannot be witnessed in the wild. Thank you. Over to Trembak now.
His master's in environmental science from Dehradun Institute. He joined WI as a master's education student and worked on Carnival Ecology Agency, Himachal Pradesh. Currently, he is working as a student associate. Over to him.
Thank you. Thank you, sir. Thank you, Dr. Bharti. So I'll be talking regarding the second segment of this talk about the preliminary results on the comparative assessment of physiological and nutritional stress in captive and wild red pandas. So for a long period of time, the red pandas have always got the attention of the scientific fraternity, not just because of its distribution being restricted to Southeast Asia, but also being classified as a mammalian living fossil, and also being more related to mustelids rather than other ursids. So it's not just an ordinary bear. So for the conservation of this extraordinary bear, aka the first panda, various conservation breeding programs had been kick-started by Darjeeling Zoo, and then under the project titled Back to the Wild, the first augmentation of red pandas to Singalila National Park initiated in 1991, followed by the second augmentation to Naura Valley National Park quite recently in 2022. So with the advancement in ecological tools, we have been able to provide a brief account of their ecology as depicted in this slide, under study themes such as habitat selection, distribution, population estimation, etc. But how are they really sustaining in the wild is still a mystery. So to unravel that mystery, a few but diverse endocrinological studies have been attempted to complement the understanding in the physiology of this animal, but unfortunately all in captivity. So, however, a comparative assessment between the captive and the wild populations of red panda still remains unexplored, and hence this study aims to fill that gap. So our study investigated by quantifying the physiological differences in red panda by using glucocorticoids, also known as physiological stress hormone, and tridothyronine, also regarded as nutritional stress hormone, by using the non-invasively collected captive and wild fecal samples, with precedence was given to the standardization of these hormones. For this study, we selected 6 individuals, namely Dilip, Kimbu, Nikki, Prasanna, Sunita, and Swati, and systematic sampling for the month of May to June this year was conducted on these selected individuals. And similarly, an opportunistic sampling was done during the same period of time to collect wild samples from Naura Valley National Park. So as mentioned before by my colleagues, a similar protocol was followed to standardize these hormones by performing parallelism and accuracy tests. And the results showed a 1:40 working dilution for the fecal glucocorticoid metabolites. And similarly, a 1:15 working dilution for fecal tridothyronine metabolites for this animal as shown here. So after standardization, the respective hormones were quantified for all the samples, and the data generated were categorized to identify differences, if any, as to in between captive and wild, and among the sexes and individuals in captivity. So after analyzing, we found that the GC levels in captive samples were significantly higher than the ones in wild, as depicted here in blue. And the T3 levels were significantly higher in the wild samples than the one in captive, depicted here in red. So we found no sex-specific differences in the physiological as well as the nutritional stress. And similarly, we observed that no significant differences were found in the GC levels in captivity, although individual variations were found as depicted in this graph. So, although we found significant differences in T3 levels among some individuals in captivity, and where T3 levels in Sunita were significantly higher than in Kimbu, Nikki, and Prasanna. So we conclude a physiologically successful augmentation at Nowra Valley National Park with the preliminary analysis showing better physiological indices being recorded from wild samples. And the captive animals were more stressed, possibly due to enclosure restrictions, overcrowding with other animals being in proximity of in the zoo, and visitor disturbances. And better nutrition in the wild individuals, well, possibly explaining a surplus food availability and also access to wide variety of food in the wild. So as we move forward, we would like to incorporate reproductive hormone profiles on these targeted animals with the ongoing physiological hormone profiles, and also assess the longitudinal hormone profiles to understand the physiological changes with respect to the environmental challenges faced by these high elevation mammals such as markhor and red panda. Also to implement a long-term comparison between the captive and the wild samples to monitor the ongoing augmentation program by the zoo. So yes, a similar systematic endocrinological assessment for the snow leopards has also been initiated with the red pandas under the same project. And we are very excited to gain more endocrine insights regarding them as well. And we both would like to thank all the members of this project who have supported us. Thank you.
All the work inter. So now I call all the inter here on the Can you take me online?
Mani, are you online?
One, are you there? So before we take one, we can take the other together. We can start.
You can start with Saurabh, please. So, Saurabh, the first question is from Dr. Bilal. He's complimenting your presentation and asks how the proposed approaches can help in future transformation and what was baseline for comparison. The comparison is between different stages of transformation.
Uh, thank you, sir, for your question. So for future translocation, uh, it can be very helpful for this translocation too, as well as for future translocation. It will be very helpful to combine both the data, both the physiological data as well as the ecological data, and that will help the administration to take the necessary decision with respect to the conservation, and which helps the animal to adapt to the new environment. And sir, in order to create the baseline, first the thing what we required is a large amount of sample and with a long duration of time. And yes, like in this study, right now the cheetahs are in captivity, but when they will be released in the wild, then we have a data of both the environment, both the captive as well as both the wild, and that will surely help us to test whether when they are in stress or when they are not in the stress, then we can compare both the data. Thank you.
Okay, the next question is from Ajinkya, 45th diploma. Please comment on possible implications of introduction of the African cheetah and not the introduction of Asiatic cheetah in India. The implications of introduction of African cheetah.
Saurabh, you want to respond? You can.
Okay. Okay. So, sorry to say, but sir, I'm not the right person to answer this question. So, you can please ask these questions to my PIs, and they will answer this better than me.
So, Saurabh, the next question is from Ashita. In hormone, nutrition is low, stress is high. As per your hormone analysis, this looks bad. What is the normal level of these hormones in the wild? Did you draw any parallel with similar studies done in Namibia or South Africa?
Sir, as this is the first intercontinental translocation study, and and also to yes, sir. So this is the first intercontinental study. So for now, we do not have any comparison for this study, but surely. Uh, Sorry, sir, I do not like thinking the correct word to answer this right now.
Thank you, Saurabh. There are many more questions for you. You can collect it in the
Yeah, thank you, thank you.
Next is for Bharti. First
Sir,
question
I'm
is
audible.
from Kaziranga. Why was there difference in gestation period of marker in captive and wild? Your opinion. And second is how long after the vivification the feces can be used for assessment? Does drying of feces affect the values?
Thank you for the question. I would like to address it in this way that once you are sitting with your binoculars and trying to observe an animal, I think it is, you can get an overall idea that the animal is pregnant, but you cannot actually find out the exact dates or the duration. For that, you always need a physiological test to check what is the actual duration. So that I think that is why there is some slight difference between the gestation period in the wild from the captivity. Because the captivity studies are actually backed up by the hormonal studies, so I could say it is more relatable with the gestation period, what we have seen in captivity. In addition, in captivity, the conditions can be a little different from wild, so which can also affect the physiological state of the animal, which could also influence the gestation period of the species. And for the second question, I will say after defecation, ideally 48 hours are considered the most appropriate for testing the hormones, and that too, once we collect the feces, we need to freeze them. And after the refrigeration process, then we start with our protocol. So that helps us to find out the hormone concentrations in the feces. I hope that answers the question.
The next is from Dr. Kushni. I think the first one you have answered with this previous question. The second one is he's curious to know where this breeding state of marker has come from.
Oh, this came from Osberg Zoo in Germany. The species that we have used in our study, it came from Osberg in Germany. So there was one male and one female that was brought from Germany. Sorry, sir?
So he's asking if the conservation breeding is going on for marker.
Yes, they are attempting to breed the markhor in captivity in Darjeeling Zoo. Yes. So now in total they have 6 individuals. So after bringing in the two sexes, one male and female, now they have interbred with each other, and now they have in total 6 individuals there.
This question is, what is the logic of the conservation breeding for German species? Because we have the same marker.
Yes, sir.
More different.
Yes.
Why not for that?
So it's like the samples which are available in the zoo. And since I mentioned in my presentation the reproductive cycles in the animals, since it's a subspecies of each other, it doesn't really make any difference if we study any other subspecies. It will definitely land up with the same gestation period or any other reproductive parameter if we study the Kashmir markhor or we study this particular markhor that I mentioned in my study. It will definitely give you the same results.
Okay, Bharti, there's one last question for you. It's from Bilal sir. He's asking what is the implication of studying hormone captivity versus wild, as they are all influenced by type of food and nutrients provided, away from the stressors available to wild populations.
Thank you for the question, sir. So, studying animals in captivity, like I mentioned in the last slide, it definitely helps you to fill in the gaps which cannot be seen in the wild. Because markhor is something is very inaccessible once it moves to high altitude heights. So it's very difficult to collect their fecal samples and to make any kind of assessment. So it is very, very important to have a batch in captivity so that you can replicate the study and see what is actually happening in the wild. And definitely the food and the other environmental condition affects the animal. That is why we could actually see some difference which was in the gestation period from the wild and the captivity.
Okay, thank you, Bharti.
Okay. Okay, thank you.
Question for member. We call the online center on Bhavani.
Yes, sir. Sir, I got. Yes, sir, I got it, sir.
Okay.
Sir, am I audible?
Yes.
Audible.
Bhavani,
Yes, sir.
this question is from Sandeep Gupta sir. He's complimenting your work and asking whether you have compared grey jungle fowl, red jungle fowl with domestic chicken.
Actually, from the collected feathers, we collected only grey jungle fowl and the RGF, and we tried to compare it with light microscope. In the light microscope analysis, we no varying degree of microstructure were observed, but as we go for the scanning electron micrograph, there looking for the plumulaceous barbule, we observed like more longitudinal ridges over the internode of spined area, and for the RGF, we mostly observed like irregular spine nodes were present, and like pronged hemuli were mostly observed in the RGF feather.
Okay. Bhavani, the next question is from Dr. Sujita. He is asking whether you have examined feather microscopic characteristics of non-Galliformes species for comparison, as this will be required for clarification of forensic samples.
Yes, ma'am. Actually, I couldn't able to hear. Can you say it once again?
He's asking whether you have compared the feather samples with non-Galliformes species. Because it will be
Actually, ma'am.
required for classification.
Okay, I got the question. So, currently, this project is only focusing on galliform feathers, and opportunistically, we also collected some passeriform feathers as well. In passeriform, when you look for the plumulaceous barb, which is like key identification feature to identify between individual orders. So, what happening is in passeriform birds, we can find crocus-shaped node. So, that can differentiate between passeriform and galliform. But when we go individual species in a particular order, that is like we need an extra reliable tool like DNA extraction method. This exact that particular bird.
Okay, the next question is you directly collected feathers from birds in the zoos, right? How effective is this process in nature when a crime had already taken place and feathers get detected quickly? Why are you not audible?
I think no, you are not visible, not audible.
Sir, now my voice.
It's now it's fine. Please go ahead.
Like when we collected feathers from the aviary, we also thought like it's impossible to collect feathers from the wild and create a feather database of it. And the question is like when we get a crime of any galliform species, so it's very hard to directly say from a degraded feather that this particular feather is from that species. But when a whole feather, particularly speaking about when we talk about pinaceous feather, there we can only rely on the morphological database. And when we look for the plumulaceous feather, we can go for the plumulaceous barbule. And that's why this feather database is created so that to match with the currently available database with us.
The second question is many Galliformes often exhibit similar feather structure in closely related species. How accurately can we ID the exact species?
Uh, we cannot only rely on the feather database for exact identification of that particular species. Further, that's why we need a DNA database to accurately say, okay, this species is matching with the finding of those galliforms. Like only when we look over the structure of spine node or ring node, it's not actually varying. So right now we are also developing morphometric measurements like the distance between the node, the internodal length, the width of the node to correctly differentiate only through the light microscopic database that will help in further identification in future.
Okay. So, Dr. Arun and Megha are complimenting you for your work. So, that's all. And there's another question, but we have sent the questions to you, and I think that you can discuss it with the people who have asked us later. Right?
Yes,
Thank
ma'am.
you.
Thank you, ma'am.
Thank you. So, for final conclusion, I will invite my colleague, Dr. Galan, for
Thank you, sir. First of all, congrats to all the speakers. I hugely appreciate all the work that has been done, and especially living in an era where human intervention and ex situ conservation is very, very difficult to avoid for conservation, and it's crucial, I would say. So, thank you for all the work and congratulations again.
particular. So we had three presentation here in this session. So all were very relevant and very nicely presented. Usually the forensic investigation of the feather microscopy, which is again going to be a new era in the science. So uh, at certain institutions level, we are not able to differentiate at genetic level whether they are domestic or wild, like red fowl and chicken. So that's why I was asking there is a relation in the morphology, but still it is doubtful because it genetic science or morphology is very but still their DNA is not possible, their marker is not possible. So such technique will be very useful, which is linked. So the next is related on the hormonal hormone. So those are also very relevant and very applicable in the giving lot of body is about the level. So gender can be added and can be used in the project which we are using, like I was in the later infrastructure project where it was not using that. If they are changing stress level using that structure, so that also can be extended there. So I am going to be very useful study in the level certification. So I consider all the gender, all the for gender their work very nicely and explain everything nicely. Thank you all. I also continue to my co-chair as well as the Lieutenant Thank you.
Thank you, Dr. Gupta, Dr. Galan, and Dr. Lee. So we'll now break for lunch. We'll take lunch is slowly. We'll break for tea and assemble here at 11 o'clock.
इनका
प्रेजेंटेशन कॉपी करना है।
हाँ सेशन 2 इसी में ठीक है। आपका कौन सा है? ये है ना आपका?
जी
हाँ नाम कर बता देंगे अपने।
मनीषा।
चला देना।
चला के नहीं देखना है, सब ऑलरेडी देख लेंगे।
ऐसा वो हटा सकते हैं एचडी में, फिर यहाँ पे देख सकते हैं।
नहीं 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 चाहिए, हो जाएगा।
देख लेते हैं आखिरी स्लाइड तक गया है कि नहीं। ठीक है।
ठीक है, मुझे वहाँ से शायद नहीं दिखेंगे ना, एडजस्ट करके। हाँ हम लोग तो नहीं देखेंगे वहाँ से। ठीक है, हो जाएगा। आप लोग का नाम क्या है मैम? इज द चेयर डॉक्टर शिवानी बर्टवाल। आपने बायो दिया है? शी इज आस्किंग फॉर दैट। शी इज। आपका नाम क्या है? मेरा पेन ड्राइव में ले ली।
अब ले लीजिए। अरे ये वाली, ये तो हो गया,
फॉर्मेट हो गया।
फॉर्मेट हुआ नहीं है, वो जो आता नहीं है
हाँ।
कि जब इसमें शायद कुछ लगाते हैं तो हो सकता है लगे। हाँ बस, मुझे लगा वाला सीन हो गया, वो खराब हो जाती है।
एक मिनट थोड़ा सा मैं
ये
नाम
वाली,
चेंज कर दूंगा।
ये वाली,
हाँ,
हाँ कर देते हैं।
चेंज कर दूंगा। सुमित प्रजापति का था। पॉइंटर है ना, चल रहा है वो माइक है, खोलो माइक पे क्या
सब रखा है कोई, सब रखे हैं लेकिन यूज नहीं कर रहा है कोई।
क्यों?
मेरे को यूज करने, मेरे को यूज नहीं कर रहे हैं ये लोग।
मेरे को हेल्प कर देना वाला लगाने के
ठीक है, ये ठीक है, इतना बहुत इससे हो जाएगा।
ठीक है,
चला के देख लेते हैं।
चला के देखो ना, ये वीडियो खाली वीडियो प्ले करना नीचे। नीचे 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 नीचे
ऐसा ही है, जूम हो गया ज़्यादा।
इधर 6 प्ले करना। ठीक, साउंड भी है ना, ठीक है, इसे कर दो।
ये तो क्या ठीक है, इसकी जरूरत नहीं है। कहाँ होगा?
थैंक यू सो मच।
चेक करना है, इनमें से ही होगी कोई।
नहीं देखा आपको सामने रहना। यहाँ पे ना ट्रांजेक्शन
किसी में कुछ वीडियो भी है?
नहीं, वीडियो नहीं, फोटोज। ठीक है,
ठीक है, ठीक है, इसको रखते हैं।
Today, this is human assessment. I invite my colleague Dr. K. Ramesh to chair the session. I invite my colleague Dr. C. Ramesh to co-chair the session and Dr. Shivani Goswami to act as the facilitator. So we have 6 presentations in this, followed by slightly longer session of comments and photographs. Slightly late lunch, so I hope you all can not be at this. Thank you very much. So welcome back to the session. I mountains to I would say microflora and then thoughts to hormonal. You can see the very diversity of presentation so far. The last presentation or the last session will be about different dimensions. I think the most interesting pieces that is human. We have a very diverse presentation. I would now request my co-chair. Good morning, everyone. To begin with, I would like to invite Monisha Asra to talk about assessing capital, policy access, and vulnerability to enhance livelihood strategies around sustainable agriculture. So, I request Shivani to introduce us.
First speaker, Ms. Kanisha Abashar, who is Master's degree in Ecology and Sustainable Development from the Tata Institute of Sciences. She is currently working as a senior lecturer in the NPCH department of Earth. She is pursuing PhD from Technology on Cheetah Park today, Kaziranga, Thailand.
Thank you for the introduction, ma'am. Thank you for the introduction, ma'am. A very good morning to one and all present here. I'll be talking about the complex interplay of livelihood capital, policy access, income, skills, and vulnerability in enhancing livelihood strategies around Kaziranga Tiger Reserve. So there is no protected area in the world which is completely devoid of human surroundings and dependency. For a long-term conservation goal to be achieved, it is extremely important to strengthen the human capital, livelihood assets, and to create a positive mindset towards factors. And for this reason, the Eco Development Committee was formed to devise sustainable livelihood option and also to have a good relation with the Forest Department and with the park management. So our basic question was to see what form of collective incentive could be strengthened for the community to elicit positive perception towards the park vis-a-vis sustainable livelihood. Our major objective is to develop site-specific strategies for aligning people's livelihood with conservation priorities of Kaziranga Tiger Reserve. So we have selected 40 villages across the 3 wildlife division. In the North Bank, which is the Bishwanath Wildlife Division, we have 8 villages. In the Eastern Assam, we have selected 22 villages, and in the Nagaon Wildlife Division, we have selected 10 villages. And in each of these 40 villages, EDCs have been formed. Our study is based on the Sustainable Livelihood Framework, which emphasizes how 5 capitals, that is the human capital, social capital, natural, financial, and physical capital, work together to enhance resiliency and sustainability in livelihood outcomes. Most importantly, it sees the interaction between the institution, policy, and structure, and how we respond to the external shocks and stress. However, this sustainable livelihood framework has failed in 2 aspects. First is to consider the emotional bond and connection with our family and how it decides our decision in the livelihood outcomes. So we have relied on the mandala framework. The research paradigm of our study is pragmatism, which is an outcome-oriented philosophical approach. The 40 study villages, there are 1024 respondents representing 189 self-help groups. Data were collected through the PRA techniques like focus group discussion, key informant interview, and resource mapping. For SWOT analysis of each and every livelihood options, we have developed parameters based on the Likert scale to determine the relative importance. To understand the livelihood vulnerability index, we have developed 4 key indicators and normalized it using the minimum maximum method. In terms of income, if we see, daily wage constitute highest percentage of income in the annual income, which is followed by the agricultural sector and livestock rearing. 5 or less than 5% to the annual income is constituted from government service, fish-based income, handloom-based income, and tea gardens. In case of SWOT analysis, we see that crop depredation by large herbivore like rhino, buffalo, and elephant is considered as the most prominent threat. In terms of strength, if we see agriculture, like mainly paddy cultivation, jute, and mustard cultivation is not only seen as an economic activity, but it is also a part of our cultural practice. Because in Assam, rice is not only a food or for our income, but it is an emotion for us. In case of handloom-based income, we see that the most important threat is the lack of marketing linkages, competition from the cheap machine-made products, and the important threat is the disruption in the supply and demand chain. In case of livestock rearing, we have identified lifting by wildlife such as tiger and leopard to be the most important threat, and in some cases policy changes, for example, like notification of new wildlife corridor has also led the people not to allow their cattle to graze inside or in the fringes of the park. In case of Aries silk rearing, we all know that it is one of the oldest practices in Assam, but due to the resource scarcity and also competition from the machine-made market, this practice is gradually declining. In case of fish-based income, if you see that protected area laws and regulation, such as the northern bank of KTR, that is the Bishwanath Wildlife Division, in the year 2008, it was declared as the 6th edition of the park. And since then, the fishing-based income was completely banned there, and people consider this as their customary rights. So this is considered as a threat. People also considered that strict patrolling by the forest department is not allowing them to fish anymore. In terms of human capital, we have seen that most of the respondents, that is 35%, have attained their primary level of education, and 8% of the respondents has no formal education. In terms of physical health, if you see that when we talk about human capital, the status of our health is very important, and diabetes and high blood pressure among the respondents is very, very prominent. Also, post-COVID vaccine had led to acute hair fall, lower back pain, lethargic feeling, and mood swings. We have also seen a gap between the skill and the engagement gap. For example, in the food processing sector, if we see that 25% of the respondents, they know how to bake cake or biscuit, pickles and things, but only 0.5% of the houses have translated this into a source of income. Similar case was seen also in the handloom sector. In access to financial institutions, we have seen that the self-help groups formed under the National Rural Livelihood Mission has mandated that each and every SHG member should deposit a minimum of rupees 50 to each SHG. Also, a corpus fund of 10 to 15,000 is given to each SHG to strengthen their institutional management capacity and build a good credit history within the group. In terms of loan, we have seen that people trust more to the private microfinance institution such as the Bandhan Bank, Swastik Bank, and also the Annapurna Bank. In terms of physical capital, we have seen that the most common form of transportation is cycle. Distance from the study villages to the NH 37 can range from 100 meter to 25 kilometer. Water is available at an underground boring of 35 feet to 80 feet. Now, there is no methodology to quantify your social capital. We can never quantify our love, hatred, or bonding. But we can see a strong networking through the concept of Namghar, which we call the community prayer hall. There is also a source of networking in the cricket club, youth club, and the self-help groups. In terms of natural capital, we have seen that 89% of the households have fruit-bearing trees inside their compound. Antiques such as bamboo and the gamhar tree is used for firewood. Medicinal plants such as the Indian pennywood, fiddlehead fern, and Leucas aspera is used to cure stomach illness and also lung infection, and sometimes it is also mixed with the fish curry to make it more tastier. In terms of livelihood vulnerability index, we have seen that in the Eastern Assam Wildlife Division, environmental factors is very prominent, such as flood, human-wildlife conflict. We can see a property damage picture by the elephant. In the Bishwanath Wildlife Division, we have seen that after the 6th edition of the park, since one stretch of Brahmaputra was declared as the 6th edition, fishing was completely banned, and this has contributed to the vulnerability index. In the Nagaon Wildlife Division, where lies the Laukha Burasapuri Wildlife Sanctuary, similar case was seen, but migration due to seasonal employment is one of the key factors contributing to this index. Coming to the policy axis, we have seen that only 33% of the respondents, they are using LPG connection received from the PM Ujjwala scheme. For maintaining law and order inside the village, there is a voluntary group called the Village Defense Party, which is formed by around 15 to 20 youth. And every night they do a patrolling with an honorarium of 2,500 per month. Fortunately, we have seen no prejudice against divorced women or any other gender-based negative norms, such as witch hunting. So we have seen that agriculture is one of the primary form of livelihood in the landscape, and across the 12 months, we have seen there is farming practices. So when we did the focus group discussion with the 37 EDCs, we found that introduction of farming equipments, irrigation, community-based tourism, and handloom-based income was given the highest priority when it comes to strengthening the livelihood options. Based on this, we have made a tentative list of farming equipments which cost less than 12 lakhs. But unfortunately, in the Kaziranga landscape, there's a practice that the eco-development hall is constructed at a cost of 14 lakhs. It's like a monopoly there, and it's like a fixed, it's like a permanent microplan template where entire 14-15 lakh is invested in this community hall where there is no hope of revenue outcome. So we have suggested that instead of investing in this hall, they can also invest in buying this farming equipments. We have also provided training to 32 individuals on travel and tourist guide with the help of RCT under the Ministry of Rural Development. After this training, 25 of the individuals have requested WII to strengthen their existing community-based models, and we complied to it. We gave them training on cultural performances, provided them with the food license, and it was established in a grand manner in the year 2022, 5th of December. But all blood and sweat went in vain because of one and only reason. There's an irrational fear among the mindset of the people that with the notification of additional areas and with the concept of tiger reserve, their land will be snatched. And when we visited the same village on 8th of December 2022, just after 3 days, we saw that nobody cooperated us, and it was completely in vain. Like our 4 years of hard work was in vain. But we didn't lose hope. We took this as a challenge, and we established another model just 1 month after it, and in the Panwari village, which is close to the 3rd edition of the park, using the same principles like, folk performances, ethnic food, guided village walk, homestays, and it's a successful model. We can see that the diversity of the visitors in this model. So as of now, around 1.80 lakhs revenue have been generated, but it's a good start, we say. To strengthen the handloom-based income, we know that we can't establish a market, but we have licensed them with the e-commerce platform. That's the pashupakshi.com, and as of now, it's a very humble start. We have generated around 30,901 rupees from the year 2023, and the pashupakshi.com has also established an outlet close to Thandavgarh, where our products are being listed. The most unique thing about our products were that we had incorporated rhino and pelican in all our handloom items. So we all know that livelihood can never be in a solo manner. So the Kaziranga National Park and the Tiger Reserve Authority has also helped us by giving the weavers a platform to showcase their product in all events such as the Kaziranga Bird Festival or the National Biodiversity Authority workshop. So in Kaziranga, we have this brand called Banashishti, which is a marketing platform to sell these products. Now, coming to the fish-based income, we all know that it is one of the most constant bone of contention in Kaziranga because people consider fishing as their customary rights. And in livelihood, it happens so that, when we talk about livelihood training and intervention, it is not to teach any new skill or introduce any new concept, but more important thing is to work on their existing skills. So you can see the picture. It was one of the most hostile focus group discussion in my life where I had to take forest guard to conduct this meeting because people were not willing to form eco-development committee and they wanted to continue their fishing rights. So we won't call it an alternative source of income, but we have incorporated and listed the concept of biofloc fish farming. And this concept has already been started by the Assam Project on Forest and Biodiversity Conservation, funded by the French government. So now coming to conclusion, we can see that addressing livelihood vulnerabilities in totality is next to impossible, considering the socio-political factors and the natural factors. However, strengthening the existing skill sets for better output is very important. Capacity building training such as health and wellness and travel and tourist guides is very necessary to meet the practical needs and the strategic needs. Most important thing is to dismantle the myths associated with the concept of wildlife corridors, additional areas, and tiger reserve if we really want to strike a balance between the conservation goal and community welfare. From my personal experience, I would say that doing livelihood project or any sort of livelihood intervention is not easy. It can never be a time-bound thing because hand-holding and systematic follow-up and bringing a behavioral change in the people can take up to more than 10 years. We have also received an appreciation letter for the government of Assam for licensing our weavers with the e-commerce platform because it is one of its kind initiative in the landscape. Lastly, I would like to thank our funding agency NTCA, Assam Forest Department, Kaziranga National Park and Tiger Reserve Authority, and all the individuals and entities here. Thank you very much.
Next, we would like to call upon Cheetah to talk about the deconstructing of our conflict.
His product is lies in understanding the biological and ecology. In his free time, he loves to do
Thank you for the introduction, ma'am. Hi, good morning, everyone. I will start with a brief introduction about my project, that is developing an integrated management plan for Asola Bhati Wildlife Sanctuary in New Delhi. Basically, an integrated management plan is a comprehensive approach to outline the long-term strategies for conservation and the management of the natural resources inside the sanctuary by incorporating all the stakeholders to gain that unified goal. So in that process, till now we have collected data on the various components. So particularly, I am going to discuss about the social, in which we are trying to means basically understand the perspective of community toward the wildlife and the authorities which are actually dealing with these conflict cases. So As part of this study, today I'm here to discuss a case study that we have conducted in the periphery of Asula Bhati Wildlife Sanctuary on the human rhesus macaque conflict. So, whenever we talk about any human wildlife conflict, there actually we are pointing out to the negative interaction that is happening between humans and the involved species. And in our case, that species is actually rhesus macaques. So, rhesus macaques are one of the new non-human primates which have the largest distribution after humans. You all have seen rhesus macaques. Even now they are sitting outside this auditorium in the corridors of WII. They are into their natural habitats like jungle and into towns and like mega cities like Delhi. So particularly today I am going to discuss about Delhi and my study site. So I will just give a brief background about that what happened in Delhi. So from 1980 onwards, we have seen a rapid increase in the population of rhesus macaques. There are many reasons behind that. One of the reasons is that we humans have negatively impacted their habitats, and we have provided them the supplement food actually which is more nutritious than what they get in means naturally. So what happened when their population increased? They moved toward the urbanized areas and caused a visible presence. People started seeing a visible presence of rhesus macaque in their colonies into the markets. So this visible presence with time changed into the negative interaction, and further on it goes to the conflict cases. And if you will see the newspapers from 2000 onward, especially for the Delhi, you will see that there are many cases of human and monkey conflict in terms of bites, in terms of means raiding the houses, raiding the shops, and even this condition started becoming worse when some deaths happened due to this conflict. So it created actually the concern among the communities, and ultimately in 2007 Delhi High Court passed a judgment in which they actually directed Forest Department to translocate these rhesus macaques from from actually main market of Delhi to the Asola Bhatti Wildlife Sanctuary. Until 2014, according to their management plan, Forest Department management plan, 18,500 rhesus macaques were transferred to that sanctuary. And in 2024, we don't have any exact data, but what forest official claim is around 26 to 30,000 monkeys. So transferring this large population to the 29 square kilometer area, it became a challenge for Forest Department to keep them inside that sanctuary. In that case, they tried to keep them inside through supplementing food, but it don't seems to be possible, and they started going into the periphery of the area, and the cases of conflict started coming from all the villages near to that area. So with that said, when we conducted our reconnaissance survey in February 2024, we observed that rhesus macaques are continuously moving in the villages and raiding the houses, and this conflict cases are happening there. And after a general talk with the community, we got to know that this conflict is actually very complex. So we decided to conduct a case study with the two broader objectives. One is to understand the perception of community toward rhesus macaques, and another is to analyze the conflict complexity, basically to identify the varying level of conflict. For this, we obtained a frame. So actually, this is our boundary of Asula Bhatti Wildlife Sanctuary, and here you can see this is the Sangam Vihar. It is one of the dense means enclosed area and highly dense area. So these photographs are taken from just 200 meters inside from the sanctuary. So you can see that what is actually happening in the periphery of the sanctuary. So for this study, we actually obtained a framework that is developed by Zimmerman et al. And we categorized our question into 5 broader areas, that is perception, conflict situation, mitigation history, willingness to engage, and views on the authority. And this framework actually works on the 3 major stakeholders and their relationship, that is wildlife and the park managers and community. So through this framework, we divided our conflict levels into 3 parts, that is dispute, underlaying, and deep rooted. So I will briefly explain what is dispute. Dispute, basically, we will say dispute when there is a visible presence of conflict, and but the community have high tolerance toward the conflict causing species and positive relationship with the managers who are trying to manage that conflict. And for the underlying conflict, this situation changed when in a long term, when managers try to means mitigate that conflict, and they perceive that conflict is mitigated, but the community who is facing the conflict don't perceive it as it is fully resolved, and this conflict it created a distrust with the managers. So when it combined with the level 1 problem, so it becomes underlying conflict, and it cannot be deal with the practical interventions. Here we have to obtain the different strategies to deal with this conflict. And for deep rooted, actually this is the situation when community started perceiving that government or any managers are willingly not doing anything to mitigate this conflict. So this sometimes becomes that due to the social beliefs and the conflict of identity. So this becomes our third level, deep-rooted conflict. For the data collection, we designed our questionnaire based on that framework only in two parts. One is to assess the attitude of people, and second is to gain the levels of that conflict, for which we use the Likert statement. And further, we conducted 200 household interviews in Sangam Vihar village. And for the analysis of the conflict levels, we use the additive scores and the composite indices. And actually to see that what factors are influencing the attitude of people, we use the decision tree analysis. With the in results, out of all the interviews that we have conducted, 49.5% were male and 50.5% people were females, whereas all the respondents mostly living from 6 to over 20 years, as you can see in the above graph. And the major group that we interviewed is from 36 to 55 age group. So frequency and impact of encounter means the community, around 88% of the respondents told they are on daily basis they are encountering with the rhesus macaques, and 67% reported that they are entering into the houses and damaging the properties like kitchen and water tanks on the rooftops, and stealing the dustbins, and even sometimes the damage to their electronics. Whereas they have impacted their daily lives in terms of property damage, disruption of the daily activities, and impacted on the farming and the agriculture on the smaller scale. And 93% of the people reported that these encounters have actually increased their workload in terms of repairing the property damages, and to spend more time on guarding the houses. So on perception about the species, around 49.5 people like the rhesus macaque, have a positive attitude, and 22% dislike the rhesus macaque, whereas 28% people have a neutral feeling toward the macaques. It actually, we found that that religious importance of rhesus macaque is one of the major factor that is influencing their attitude. For those who have that religious connection with the macaque, around 49% have a positive attitude. They like the rhesus macaques, whereas the aggressive behavior of macaques are actually influencing in a negative way. People who have faced any kind of aggressive behavior, they dislike the rhesus macaques. And for those who think they are cute and intelligent, means basically go in the appearance, for them this is like they don't like also, they don't dislike also. It's like a neutral feeling toward the macaques. And the religious importance have also influenced the feeding activity of humans to the rhesus macaques. Upon asking this question, around 90% of the people have fed rhesus macaques in their lifetime. And when we ask this views that shall this penalty should be there on feeding rhesus macaques, so 71% of the respondent said they oppose the feeding of rhesus macaques. So on further going, we were trying to understand that how severe that community perceived this conflict is. So 64% of people perceived this as a severe conflict, and whereas only 10% people consider that it's a mild issue. And when we ask that what they think is the cause of conflict, so 47% of the people told that it's maybe the garbage dump that are inside the or in the colonies that are due to which this conflict is happening. And even a majority of the people, like 30.4% people said due to the lack of food and water resources, they are coming into the colonies and causing the conflict. And around 76% people perceive that this conflict will increase in future. And 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 upon asking what measures they actually suggest, and they think that will work on the rhesus macaques, what community thinks. So they told they actually answered more on the sterilization and translocation of the macaques, but how effective these methods are, that question is still means I think everyone is looking for this answer. So yeah so yeah, so one of the broader question is to understand that are the community aware about any kind of efforts that are going on to mitigate this conflict. So majority, it's like not majority, or almost 91.5% people suggested that they are not aware of any kind of mitigation efforts that government is doing for that. And they are also not aware of any kind of helpline number or any reporting channel where they can report about the conflict cases of this human wildlife conflict. So and whenever, if in such a way they can they in somehow they respond they report that conflict case, 67% believe that department responds very late. And even like 25.1% people suggest they sometimes even don't respond to their complaints. So we on the further analysis we found that that response of the government to these cases and the cases is actually one of the significant factor that is influencing that future perspective of community toward the conflict. And even there are other major factors also, like the cultural importance and feeling toward macaques, and also the age of the respondents. And for the last one, that when about the willingness to participate in the management efforts or any kind of conservation efforts that the stakeholders are other stakeholders are trying to do. So only 21% of all the respondents were agreed on the fact that yeah, they will contribute to such kind of an initiative. And but the majority, like 62% people disagree that they cannot they will not participate in any kind of mitigation measure. So when we asked about the reason that why they think like that, so around so two means major responses we got. One is that around 28% people think for macaques there is no place to go now, especially in the Delhi. This is specifically for our field site. So doing any kind of mitigation will not work. But there are 25% people who think forest department is not working honestly, and or the managers who are dealing with that thing, they are not working honestly. So means going in that factor will be a little difficult. And here also we found, okay, sorry, I will go fast. Okay. So finally, for the level of conflict, we found that when we conducted this additive scores and overall composite indices, we found that the level is falling to the underlying conflict, which actually means that to deal with this issue, we have to build up a relationship with the community rather than just focusing on the practical interventions. Sorry, just I will take 1 minute. So the most biggest challenge that why we are presenting only macaques, not on the other components, is that Sangam Vihar is actually the encroached colony that what government claims, and there is a distrust between forest department and the community. And one of the important factor that the forest department is feeding rhesus macaques inside the sanctuary in a large scale. So and they are spending, according to this data is according to 2014, that they are spending around 1 crore just to feed the rhesus macaque without any sterilization or any other population control method. So with that said, I would like to acknowledge all these peoples, and thank you for listening. Thank you.
Next, we would like to call upon Gaurav to
A very good morning to all of you. So I'll be directly asking you a question in the beginning. So what are the 5 things that you think of when you think of Delhi? What are the 5 things that come to your mind? So a lot of things, obviously. So most of you might have thought about these things, new parliament building and other things. But unfortunately, today we are not going to talk about any of these in this presentation. We are going to talk about mammals from Delhi. Okay, so there are 190 mammal species which are regularly observed in urban areas worldwide. So that leads us to our presentation, mammalian ecology in an urban context. So understanding the mammalian ecology in Delhi. So this is our project and the information that my friend Mukesh already told you about. And the 2 components that I have been mainly working on are birds and mammals component, but right now I'll be mainly talking about the mammals component right now. So let's first try to understand our study area. Then you will be able to relate the outcome, the results in a much better way. So for that, I would like to tell you a bit more about our study area. So as you can see, that there is New Delhi on that side from Sanghavihar side, which is a encroached colony, and towards this side, the eastern side lies Haryana, and on the northern side as well. And in the middle of the our study area, there is a big village, Bhatti village. So I want you to just try to have a look at this picture. The middle parts of the sanctuary, they have ridges and mines. So that will later help us to understand the ecology of mammals in a better way. So I'd like you to have a look at it. So now I would like you to have a look at this map, which shows the intensity of the roads in our study area. So you can see there's roads almost everywhere. There is very few small patches where there are no roads, and the yellow portions, they show the roads that are 100 meter within the 100-meter range. So these are the water holes in our study area. There are very less permanent water bodies. So these are water holes where the water holes are actually scattered. The blue dot there at the end, it's Neeli Jheel. Neeli Jheel is very famous in Delhi, and people go there, and it's a very, very beautiful lake. So this is LULC and NDVI maps. As you can see that it's mostly a precipice-dominated forest, and there are red patches in between which indicates the village. So now the question arises, what makes Asola Bhatti such a unique wildlife sanctuary and as a study area? The first one is that it's located in Delhi, obviously, and it's the only wildlife sanctuary in Delhi, apart from some steep parks and bird sanctuaries such as Okhla Bird Wildlife Sanctuary, and it's part of Aravallis, which is one of the world's oldest mountain ranges in the world. The second thing that makes it unique, there is no natural water resource in the sanctuary for animals except a Neeli Jheel, which is very steep, which has actually been created because of the deep mining. So it's very deep. People say that it has almost touched the water table. Another interesting feature about our study area is there is a big 10-feet wall between the forest reserve of Haryana and the forest reserve of Delhi. So it divides the same uniform wildlife area into 2 parts, which is not good, obviously. And then there's a big village in the middle of the sanctuary, the Bhatti village, which was actually created in 1980s. These people used to mine there, and then the people settled there, and the village was created. Okay, you won't believe this fact. Delhi Forest Department feeds around 8 lakh rupees worth of food to monkeys. And trust me, the bananas that Mukesh showed you, they were of much better quality than what we used to get in the market. Okay, the sanctuary has also been divided into enclosures. There are enclosures for plantation sites. So to protect the newly planted saplings, Forest Department has created the enclosures. There are mines because the area was being used as a mining site in 1980s where for Badarpur sand, which is very famous there, and people used to mine it there. So because of that, there are mines in our study area as well. The most risky one, illegal cross-border liquor trafficking between Haryana and Delhi. So people from, not people, I'll say, I need to be careful. Some people from Haryana, they take illegal liquor into the Delhi and sell it at higher prices. So we were always advised to not go to the study area late night, but obviously we had to. Okay, there are very limited studies, despite being in Delhi, in the national capital territory of Delhi, there are really limited studies available. So what were our objectives? What we wanted to find? The first thing, obviously, since our project is a management plan, so we wanted to know what mammal species are present there, and how are they distributed spatially as well as temporally, and how are these different mammal species interacting with each other, both spatially and temporally. So what are the different ecological and environmental factors that are affecting them in such an urban area? So there are plantation sites. So we also wanted to know, do they favor old plantation sites, the mammals, or the new plantation sites? Also, we wanted to understand whether the animals are crossing the wall between Delhi and Haryana. So methodology, we conducted camera trapping in May to June. So 28 camera traps were used in May to June, which was very hot at that time, and it's especially more hot when you come from a place like Kashmir. And then the data was cleaned and processed in ExitPro and CamTrapR and with a 30-minute interval time. Later, our data was analyzed in Excel, ArcMap 10.8, and RStudio. So this is the location of our camera traps. We had to be very careful to not to install the camera near a village, because it otherwise would have been okay. So the methodology part, we also calculated the replace K function to understand whether the animals are distributed, they are clustered, they are randomly distributed, or they are having a random distribution. And at the same time, we run generalized leader modeling to understand the relationships between different variables. And Priyanka's niche index was estimated to calculate the overlap between different species. So for the results, the relative abundance index shows that the macaques were the most common animal species there, followed by stray cattle, cows mostly, and sambar is the animal which is found least. There are a total of 22 mammal species we captured in our camera traps. Obviously, there are more because we didn't bush rats and other mammals like shrews. We didn't see them in our camera traps. So this is a picture from our study area, and we further subdivided the relative abundance of cattle. So cows were the most common stray animal type, followed by dogs, and there are camels too there, okay? And after that, our species accumulation curve shows that we had, the sampling effort is sufficient because we got good number of species within just a week almost, and we have a flat curve. So now the question arises, how are these animals coexisting at spatial and temporal levels in ABS, WS? Because this is such a small area, so obviously it's very difficult for different species to avoid each other, but there must be some way by which these animals are actually existing together. So for leopard, as you can see, the central regions and the southern regions are having the most density, because the northern and at the bottom, it's the place where Bhatti mine is present. So that's why there's least disturbance. The leopards are present in good number. And for the activity overlap, we compared it with humans. As you can see, the leopards are showing a crepuscular activity pattern. They are mostly active during early in the morning and late in the night. And there is very less overlap between humans. And so leopards are actually avoiding humans temporarily, but not spatially maybe. And for the distributional patterns using replace K function, we wanted to understand how are these leopards, whether they are clustered, whether they are randomly distributed, whether there is a proper dispersion. So there is great level of dispersion, which is understood by the fact that there are very small areas in the sanctuary which are not disturbed. So for the striped hyena, as you can see, striped hyena and leopard almost overlap each other spatially. And for the humans part, they are mostly nocturnal. So they are avoiding humans temporarily. At the same time, there is a very high replay K function value, which indicates that there is high level of clustering at some particular places, which in this case is actually our mines, because hyenas were present in mines in good numbers, like not in huge numbers, but in very basic numbers. So for the jackals, they are having a clumped distribution all over the sanctuary. They are present almost everywhere, but they are not avoiding humans. They have become used to humans actually, which is indicated by our activity overlap. So for the replace over function, it's again showing clustering for golden jackal. So almost all the animals, they are showing the clustering. So this rhesus macaque, for this pattern, there are actually feeding points all over the sanctuary along the road. So those feeding points are the places where the rhesus macaques are mostly present. That's why the map is showing such a clumped, hot heat map. So this is the distributional pattern of macaque. They are also showing clustering, which is understood by the fact that there are feeding points in the sanctuary. So for the nilgai as well, it's found everywhere with a slightly different distribution, and it's more in the zone A. So zone A is the part above. So it's not avoiding humans. So they are having good interactions with humans actually. So they have they show a stepped increase, which means that there is good number of clustering in this species at distinct distances. So for the spotted deer, it's found only in zone B at specific places. So we did not find a single spotted deer in the first zone, in the above zone of the sanctuary, which is actually very much disturbed. So for the stray cattle, they are found everywhere, and they are not avoiding humans. So there are also clusters of information present. So as you can see this image, the dogs and jackals, they are roaming around together like friends from our camera trap. So the stray dogs also show, the stray dogs, they are not avoiding humans as indicated by the map. Similarly, as you can see this image, this particular pig had 12 babies actually, piglets. So they are also having a clump distribution, and then they are having the nocturnal. As you can see, they are mostly active during night. So the peaks at in the midnight after 6 at 12. So they are again showing the clump distribution with the stepwise increase. So for porcupine too, they are avoiding humans. They are having the nocturnal patterns. So this is the sambar. The particular interesting thing about sambar is that it was only found in the last Bhatti mine region. We did not find sambar elsewhere in our area. So we have very limited data for this sambar, so we will need to collect more data to understand the temporal patterns. So there's a question arises now, why are almost all animals mainly distributed in the central and southern regions of the sanctuary? We asked this question to ourselves, and possible explanation for this is that, as you can see, there's village from that side, there is road at that side, and at the Faridabad Gurugaon highways there, which offers a great level of disturbance to animals, because of which the central and the last region, they are having connectivity to the forest reserve in Haryana, because of which animals are maybe feeling more safer. So there have been around 10 leopard deaths in the last road there. So there is a high need for creating an underpass there, which will actually help us reduce the number of these. So there is also spatial overlap between leopard and its prey species. You can see leopard is having the maximum spatial overlap with nilgai, followed by wild pig. For hyena, it's mostly spatially correlated with stray cattle. So for herbivores too, wild pig and stray cattle, they exhibited the highest level of overlap. Similarly, the highest overlap, we also divided the herbivores into small, medium, and large. The highest overlap was shown between similar sized groups. So for the Pianka's niche index, as you can see that the dogs and golden jackal, they have moderate niche overlaps, while for the observed versus expected co-occurrence of species, as you can see, dogs and jackals showed higher observed occurrence than what we expected, because dogs and jackals are kind of roaming at water holes together. So for the herbivores too, as you can see, cattle have more data points. They have good overlap, resource overlap with other species. So how are various animals utilizing both new and old plantation sites? So this is the data from the old plantation site. As you can see, it has good diversity. A lot of animals were seen there. Stray cattle was the mostly seen animal. And for the new plantation site, we saw very few animal species, which might indicate disturbance there. So it indicates for the management plan perspective that old plantation sites maybe serve as good habitat for many mammal species. So we wanted to know, do the animals cross the wall? So we installed a camera at the wall. Yes, sir. So as you can see, the animals are regularly using the wall. So the Neil guy is coming to Delhi, and I think this Neil guy wants to go to Delhi, and thank God the wall is broken for him, because otherwise he would not have been able to pass. So how do animals respond to major disturbances temporarily? As you can see, we plotted three main disturbances: stray dog, humans, and vehicle. So all of them are, Neil guy is not avoiding any of these three disturbances. So for the leopard, as you can see, the leopard is avoiding all the three forms of disturbances, whether vehicle, human, or stray dog. Similarly, for spotted deer, it's not avoiding humans or other disturbances, but the overlap is very less. For the hyena, as you can see, there is a opposite direction. It's very nocturnal, so it's avoiding all the forms of disturbances altogether. So the wild pig too, it's nocturnal, so avoiding major disturbances. Ruddy mongoose is not avoiding. So how are different ecological and environmental variables affecting different mammal species? So we performed GLM on it. We took such variables. So first thing we had to do was check which model fits best. Sure, sir. So this is the model that we ran, and then you can see what different variables affect what species in what way. So this is the correlation matrix for leopard, which tells us about different variables affecting the species at one point of time. And then we wanted to also know how the different variables or covariates are affecting the leopard capture. As you can see, there is positive capture for certain variables and negative capture for others. So for the hyena, we did the same thing. We had to check the best model. So we ran a negative binomial model. As the variance was greater than mean, so there was great over dispersion. So for all species, it's not possible to show for all 22 species, but I'll just limit myself and restrict myself here. So this is for spotted deer correlation matrix and different covariates. So limitations, we have a small study area, single Caesar data. So there are zone 2 and zone 3 are having the most animal records. So this is a picture from the denning of jackal. So what we want to do next, we want to conduct extensive camera trapping and also investigate potential hybridization between dogs and jackals, examine denning patterns of jackals, conduct a more detailed study of mines in ABWS, and assess the impact of fencing on the ecology of mammals, verify the presence of rusty spotted cat. We are highly sure that rusty spotted cat is present in Delhi. It's just a matter of time it will be seen there. We did not get it in camera trap, but we hope people get it next. So these are all the people who helped me. I thank every one of you. So thank you, and be eco-friendly.
The next talk will be on the development and community engagement. We will talk about biodiversity conservation. The speaker is Kalpana.
Ms. Kalpana Roy holds a master's degree in environmental science. In 2019, she joined the Biotech Institute. She is currently working on a project which aims to identify and measure agents of human wildlife conflict in Chhattisgarh, Jharkhand, and Assam.
Thank you for the introduction, ma'am. I'll be starting like this. In India, elephants are celebrated in every way imaginable. They are the gods in our temples, symbol of wisdom in our stories, and showstopper in our festivals. They aren't always the gentle giants. In some spaces, they have developed a taste for playing a different role. Imagine this. One moment we are leaving banana as an offering to Lord Ganesha, and the next moment you are being chased away by a tusker who has been helping himself to your entire banana crop. We live in a dynamics where we build temple for elephants on the one hand, and we are building fences and trenches to keeping them away from our villages and farms. This brings us to the topic of today's presentation, that is pattern of human mortality by elephant over 2 decades. By this study, we aim to understand the trend and dynamic of the incidents. Before diving into the pattern, let's look at the number of the elephant distribution and root causes. In India, India holds nearly 60% of Asian elephants. India holds nearly 60% of Asian elephants, nearly 30,000 in numbers. Their distribution is in northern, northeastern, central, and southern region of India. And southern India hosts the primary elephant population of India. As human population expand and encroach on elephant habitats, these gentle giants often find themselves in struggle for space, leading to tragic outcomes for both sides, often resulting in crop damage, property destruction, injuries, and loss of life on both sides. Let's talk about the causes. Causes include habitat loss and fragmentation, that is deforestation, agricultural expansion, infrastructure development of roads and railways, human encroachment, settlement near elephant habitat, expansion of villages and towns, elephant behavior, their seasonal migration pattern, attraction to crops like sugarcane, maize, rice, etc. And then comes human activities, illegal logging, poaching, and retaliatory killing. Climate change, environmental stress, changes in rainfall pattern, availability of natural forest and water. Every year, elephant causes more than 500 human fatalities in India alone. If we look at the numbers started changing in the year 2019, which makes the last 5 years more intense. We took three states for our study: Assam, Chhattisgarh, and Jharkhand. The data was collected from 21 forest divisions of Assam, 19 forest divisions in Chhattisgarh, and 22 forest divisions of Jharkhand. We segregated the data year-wise, division-wise, season-wise, and gender-wise to see the patterns, and we also took out the names of the villages from the data and the location points of the incidents all over the states. This is the framework for land use land cover classification. We did land use of all 3 states from 2000 to 2024 using random forest algorithm in 5 years interval to see the changes in the landscapes. Coming to the results, in 23 years, 3 states, 62 divisions, 4,000 total incidents were 4,377, which includes 3,547 deaths. These are the map of LULC of 3 states. We can see that in all states, the agriculture and settlement has increased, ranging from 50 to 91%. These are the hotspot maps of all states showing the parts of the states where the incidents have happened in most of the numbers in all states. This is showing the year-wise distribution of incidents in all 3 states. In Assam, the total number was 1,807, which was peak in the year 2018. In Chhattisgarh, the total number of incidents was 828 and the peak was in the year 2019. In Jharkhand, the total number of incidents was 1,740. The peak was in 2009. This is the divisional distribution of the incidents in all three states. In Assam, Golaghat, Udalguri, Sonitpur East, Sonitpur West, and Golpara had experienced the most number of incidents over the years. In Chhattisgarh, Jashpur, Dharamjaygarh, and Surajpur had recorded the most number of incidents in all 23 years. In Jharkhand, Ranchi, Khunti, and Saraikela experienced the most number of incidents in all 23 years. Comparing all the states, monsoon season showed the highest number of incidents in all three states. If we talk about the ratio, for every 10 human deaths, 7 of them were male and 3 were females. This map is showing the extent of human deaths and injuries in Assam from the year 2000 to 2023. We categorize the villages as having high, low, and medium incidence. 5 villages had the incidents over 30 in 23 years, and in 22 villages had the incident ranging from 15 to 30, and 500 villages the incident were ranging from 1 to 15. This tree map is showing the number and the name of the villages where the incident have happened in the maximum numbers over 23 years. Similarly, in Jharkhand, 4 villages had experienced more than 30 incidents over the years, and 5 villages had experienced incidents ranging from 50 to 30, and 318 villages had experienced the incidents from 1 to 15. And the tree map is showing the names and the numbers of the villages having more number of incidents. This is the map of Jharkhand. In Jharkhand also, 3 villages had the maximum number of incidents ranging over 30. 16 villages had the incidents ranging from 50 to 30, and 457 villages had the range from 1 to 15. And there's also the tree map for showing the name and the number of incidents of villages. We took 7 variables to see how these variables are affecting the incidents happening in all the states. The variables were road density, railway density, waterway density, forest cover percentage, crop cover percentage, and built-up area. 4 of them showed the variance in incident and non-incident side. In road density, villages with higher intensity of human death had higher road density across the states. In railway line density, villages with higher intensity of human death had higher railway line density across the states. This is for Chhattisgarh and Jharkhand. In Assam, there were no points near the railway lines, so the graph was not coming right. This is the percentage crop. The villages with human death incident had higher percent crop cover and less variation in Assam and Jharkhand, within 15%. The pattern was reversed for the state of Chhattisgarh in terms of crop cover, but the variation was same. This is the forest percentage. The villages with human death incident had higher forest cover and less variation in Chhattisgarh and Jharkhand. The pattern was reversed for Assam in terms of forest cover. Summary. From this study, we can conclude that Assam holds the highest number of incidents in last 23 years. As in Assam, the total incidents were 1,807, and in Chhattisgarh it was 828, and in Jharkhand it was 1,740. After segregating all the data, as we focused on the villages, so we found that villages with high number of incidents are situated near the forest areas. Linear infrastructure density was found to be high in extreme conflict villages. Monsoon had the highest number of incidents in all states. As I said before, there were more male than females, and the ratio. Top 10 conflicting villages represent 32.7% of human deaths in 3 states. Forest cover, crop cover, and intensity of linear infrastructure contribute to human death and injury in 3 sites. Need for site-specific mitigation measures, and based on the intensity of conflict, prioritizing the villages with high number of incidents should be done in future. I would like to thank all these people. Thank you.
Thank you, Kaziranga, for mixing before our time. And next speaker is Kaziranga. Is going to talk about the island itself, mapping Dugong hotspot using science.
The last speaker, Mr. Cheetah, holds a master's degree in environmental science from Dugong University. For the last 5 years, he's been working on community-based hormonal modulation in Human-macaque.
Thank you for the introduction, ma'am. So today I'm going to talk on how we utilize the new approach to study dugongs in the Andaman Nicobar Islands. The quote that is written here is simply paying tribute to the special group of people who are helping the researchers like us to make a difference. So we call them as a citizen scientist. As many of you are aware of about the citizen science, so it is a tool that has been adopted by many scientific community who approaches the local people to study the nature in a better way. There are multiple citizen science platforms that are available globally as well as in India. There is an increase in use of citizen science tool in the marine field as well, because of certain limitations that we face while collecting the data, like limited manpower, limited funding, and the larger geographic area. That is because many marine researchers have been using the citizen science field to collect the data. As you are aware, all the citizen science platform which are available globally are the internet driven based. So to collect or extract the data, it requires an internet. So what if you are working in the remote island of the Andaman Nicobar Island, which itself is a very remote, and forget about the getting internet connection, even the cellular network is not available in most of the places. In addition to that, the species that we are focusing on for the study itself is a very elusive species, very rare to find. So welcome to the world of dugongs of Andaman Nicobar Island. So before going to the topic, let me brief you about what the dugong is. So dugong is the only herbivorous marine mammal, which are also known as sea cow, because they primarily and only feeds on seagrasses. As you can see in this video, they are also known as the farmer of the sea, because the way they are feeding on the seagrass meadow helps in mixing nutrient in the sea, which automatically helps in seagrass growth. And seagrass also act as a nursery ground by providing the shelter habitats to the commercially important fishes, which is why dugong holds a very important ecological and important economic function. That is why saving them is a very crucial thing. Globally, dugongs are found in 42 countries, in which India is one of them. And in India, they are found in Gujarat, Tamil Nadu, and in Andaman Nicobar Island. And in the islands, dugongs holds a very special importance because it's a state animal of the islands. And locally, they are called as pani suar. To conserve dugong, first we need to know where to find them and what are their distribution hotspots. So while identifying these their hotspots, we face several challenges, like the geographical area of the islands, which is around 2,000 kilometers of coastline, and the population itself is a very low. So chances of sighting Dugong directly is very low, and also there are certain logistical constraints like unable to reach the difficult accessibility to reach the area, and there are certain restricted area which are not accessible for researchers. To overcome these challenges, we have reached out to the citizen scientists, which are our local seafarers of the islands. So for that, we have created a citizen science initiative program called Dugong Monitoring Program. So for that, we have tried to get the sightings from three media. The first one is boat-based sighting, from where we have reached out to the fishers community, petroleum-based agency,
solid egg मिला लेकिन ना
and the boat operators. So that's how we got the boat-based sighting. And for the underwater sightings, we have reached out to the scuba divers, and they have been providing the underwater footages of dugongs. But you can see here in the video, the diver and dugong, they are swimming together here. And for the aerial-based sightings, for the first time, we have reached out to the defense bodies, and we have involved them in the monitoring program, which is the Indian Navy and Indian Coast Guard, and they are providing the aerial-based sightings from the different altitudes. So before involving them in the monitoring program, we have done the massive number of awareness program with our respected stakeholders, like the fishers folks and the petroleum-based agency. So where we have briefed them about what are the dugongs and why they are important to us and why we have to save them. So this is how we have approached the stakeholders and involved them in the monitoring program. And also we have trained them to identify dugongs in the different areas, like for underwater sightings, boat-based sighting, how dugong looks, and for the aerial sightings. And for the first time, we have been able to engage the Indian Air Force in our dugong monitoring program. So we are hoping to get the dugong sightings from these agencies also. So how the data was collected? So as you are aware about the citizen science platforms that are globally available, are mainly internet-based platforms where it requires the internet. But in the case of our field sites, where the internet is not available, so getting the sightings itself is very difficult. So how we collected the data, like, so first we did a monthly phone follow-up call with the stakeholders like fishers and forest petroleum-based agency on a monthly basis to get sightings. Secondly, there are some active informants who have been providing the dugong sightings very actively, like defense bodies, scuba divers, and lifeguards through WhatsApp media. So based on all the effort that have been put by covering all the geographical area of around 2,000 kilometers of coastline, by sensitizing the 8 different stakeholders, we have got the huge data of around 615 dugong sightings. And based on these sightings, we have been able to map the hotspots of dugongs in the Andaman Nicobar Islands. So if you see this, if you see the area that are in orange color, from where we have received the more number of sightings, which is the Richie's Archipelago and South Andaman region, it is because all the stakeholders were available in this region, and accessibility to this area is very easy as compared to the other group of islands, like North and Middle Andaman and Little Andaman. And also, we have got the sightings from data deficient area, which are not accessible for researchers also, like North Sentinel Island, where the Sentinelese live, and the Flat Bay Island, which is a part of the Jarwa Tribal Reserve area. So these were the areas where always a data deficient area, but with the help of defense body, we have been able to get the sightings from this area also. And also the sightings of carp, which suggest there are active breeding population of dugong in the islands. And the most important and interesting is that got of the herd sighting that have been reported after 8 years, 40 years. Also, that we got the sightings from the area where it was thought to be locally extinct after the tsunami, which is the Little Andaman and the Great Nicobar Island. And also the lower number of sightings that we received from the Nicobar Island, it's because that is the area that we have, that is the new area we have explored in terms of Dugong monitoring program. And also because of the geographic isolation, and also our stakeholder distribution also get reduced because it's a tribal protected area. If you see how the stakeholders have contributed to report the Dugong sightings, the fishers, tourism, and the defense bodies were the most active informant throughout the island groups. So here I would like to draw your attention here particularly. So because we have got an interesting trends in terms of reporting region-wise Dugong sightings by our stakeholders. So if you see the blue bar, which represents the fishers community, they were the most active informants, followed by the defense body only in the South Andaman region. And in Ritchie's archipelago, the scuba divers were the most active informant, followed by the least number of sightings that we received from the Nicobar Island. That is only 12 sightings. As you can see this map, how the sightings were distributed that were reported by the different stakeholders. The fisher was the most active informant in terms of reporting Dugong sightings, because fishing is the primary occupation in the island groups. And in defense body, the most of the sightings were restricted to the South Andaman region, because this is the area from where they start the aerial patrolling, and this is the main headquarters. This is because we have been getting the most sightings from this region, from defense body. And from the tourism sector, we have been received the most sightings from the Ritchie's archipelago, because this is a very tourist hotspot where people go for diving, and there is a very flourished scuba diving industries, and they have a very designated diving spot. That's why we have been getting more number of sightings from this region by scuba divers, and few sightings from the North and Middle Andaman region that were only reported by lifeguards only. We also got an interesting trend in terms of reporting sighting month-wise. So we have got the lowest number of sightings here from May to October month. It's because this is the month where the cyclone and monsoon is very frequent. So this also becomes a very limiting factor even for our stakeholders to carry out their routine work. That is why we have been getting very less number of sightings. Also in this month, even our cellular network gets also disrupted. तो कॉल लगता ही नहीं है, तो सेटिंग्स कहाँ से मिलेगा? Let us come to the nature of sighting. The solitary individuals were the most commonly reported throughout the island groups, followed by the pair individuals, which were composed of both the adults or the adult and calf in the group, and the sighting of herds. So I would like to draw your attention particularly in this photo. So where you can see the one dugong which is disguised in the corals and rocks. So this sighting that was shared by the Indian Navy. So we, with our capacity building program, we have been able to train them how to identify the aerial sightings. So this is the change that we are seeing in the defense body while reporting the dugongs, that they are able to identify dugong in the sea. And coming to the herd size that we have received after 40 years. So we have categorized the sighting that we have received about herds into two categories. The first one is smaller herds, which includes the 3 to 5 individual. So you can see the 40% of the sightings about smaller herds is a composition of only adults, while the remaining 60% population were the composition in which the calves are present. And the larger herds, which include the 6 to 13 individuals, in all the sightings, calves were present in the groups. So if you see this map, most of the sightings of larger herds were only from the South Andaman region, which is a restricted area, sheltered area, where even fishermen can't go for the fishing. And also these sightings were only reported by the defense body because they get the larger surface area while doing the aerial patrolling. And also we have got the maximum 13 dugongs were sighted in the one single herd. That was around 40 kilometers offshore from the one small island that is called Sink Island. And also we were able to get the sightings of herds, and the first time it has been photo documented by the defense bodies, and this is the first time we have got the sightings of herds in the lockdown time. So based on the underwater footages that we have received from the scuba divers, so with that footages, we have been able to identify 3 individuals based on their body marking, which are frequently visiting this area of Shahid Island, which are showing the site fidelity towards this island. Sorry. We also got the interesting observation about the natural history, where you can see the one dugong, the one identified dugong is showing the potential active sexual phase. So, in terms of citizen science can also be helpful in observing the natural history also. And the sighting of larger herds that we have received from the defense body and the fishermen, at the anecdote observation that was shared by the fisher folks and the defense body. So they were saying all the calves were in the middle in the group, surrounded by the adults, which was showing the parental nature. So, based on the result that we got, just one minute. Based on the result that we got, we have been able to identify the Dugong hotspots area, and it has also benefited us in both way, because it also enhances the local awareness of the local stakeholders, which we are sensitizing towards. So it automatically helps in the species conservation also, and herds sightings that have been reported after 40 years, and also the data sightings from the data representation area, which were completely not known before that. Although there are certain limitations of the citizen science, because as I mentioned earlier, there was the unequal stakeholder distribution in the islands while reporting the sightings. So this will always be there, because in reporting sightings, and also there was a sampling bias while getting because the last area that we explored in terms of Dugong monitoring program was Nicobar Island, from where we got the very less number of sightings. So this sampling bias is also there, and sometimes also volunteers also lose interest in terms of reporting sightings. That is why we have to re-engage the community again and again to have a good rapport with them. Also, it also enhances the knowledge about the species towards communities. So based on that, I would like to thank the following people, and special thank I would like to give our Dugong heroes. Without their help, we would not be able to get the Dugong sightings, and we would not be able to get the hotspots area. So one more, the important slide that I would like to. So on behalf of our team, we would like to pay a special tribute to one of our most important informant. We recently lost him in line of duty in Gujarat. So Sir Karan Singh, still holds records of reporting sightings of maximum Dugong sightings from the Coast Guard. And without his sighting report, we would not be able to get the sightings from data deficient area. So we dedicate this talk to Sir Karan Singh. Thank you.
For the technical session. I request all the speakers come to the stage. And also, we request all the speakers to be very precise to answer all the questions. We have received so many questions. Instead of beating around the bush, please be the answer very precise. First, I would like to call upon Manisha. The first question by Dr. Kaziranga, and he has commented on excellent work. All 37 PDC are joined whatever reserves are functional? And is there any linkages between EDC and self-help group in the same page?
Thank you for the question. For the first question, are all 37 EDCs adjoining Kaziranga is functional or not? It depends. In Eastern Assam Wildlife Division, around 9 EDCs are functional, and in the Nagaon Wildlife Division, 3 EDCs are functional, and in the Vishwanath Wildlife Division, only 2 EDCs are functional. And to make every EDC functional, we had also conducted intensive capacity building training workshop. And is there any linkage between EDCs and self-help groups in the same village? So actually EDC and SHGs are very different local institutions, but yeah, for example, it's not directly linked, but when we give training through EDCs in terms of strengthening the livelihood capitals, it is always the SHG members. So the beneficiaries are linked, but in concept, these two are completely different entities. Yeah, okay.
The next question is by Prashant, complemented your great work. Have you tried to predict the behavior using the theory of planned behavior?
Yeah, thank you for the question. Actually, theory of planned behavior, we largely use it to talk about the individual behavior, but here in addressing the livelihood issues, we are talking about the collective effort. So in livelihood aspect, we haven't addressed the theory of planned behavior, but yes, to understand the behavior of the heads of the local institutions like Eco Development Committee and Village Defense Party, we have used the theory of planned behavior. And during my last year presentation, my topic was on this theory of planned behavior, where we said that sense of belongingness towards Kaziranga plays is a very important normative role when it comes to developing a conservation mindset.
Thank you, Nisha. That's all for you.
Thank you.
Next question is for the computer channel. We have so many questions for the face chat. So, the question asked by Dr. Ramkumar. What do you think is the contributor of indirect poisoning, that is garbage disposal? And the second question is, if you attempt to quantify the conflict in economic terms, example, people may have to invest in installing grills in their houses to prevent monkey raids.
Yes. Sir, for first question, actually, this indirect poisoning is one of the major cause of that we believe that they are getting food rather than just like I gave an example ki forest department is feeding, but there are a lot of groups of macaques that are roaming into the near to the means periphery and other areas where they are getting indirectly poisoning either, and most of them are from the garbages only. And for the second question, sir, yes, actually they are facing an economic like conflict. Yes, and they are actually when they are like means when they are facing the conflict, so they the property damage, they have to invest a lot of their economy or the money that they are getting as a salary to repair that damage, and they are using like this fencing, this steel fencing, and I think you have seen the cage that sharp metal fencing over the tanks that people keep. So they are investing in that things. Yes.
Second question is from our researcher, complemented the nice presentation. From your understanding about human-macaque conflict in the current studies, what control measures you can suggest to reduce recent macaque conflicts at WIA?
It's actually very hard question to answer. Means I was thinking someone is going to ask this question. So, if overall, if you will see the human rhesus macaque conflict, basically what I think it's little bit difficult because macaques are very adaptive to the measures. They are one of the primates who are like have that intelligence and that learning behavior. So like means I don't know I can say that or not, but like we are using that sticks and other thing in the WI right now, and I believe that it's not going to be work in future. So we have to look on the broader things to deal with this conflict, like reducing maybe the like intervention to reduce the population in terms by doing the sterilization or the oral contraceptions that we can give, and other things like managing the garbages and including the municipalities of the Dehradun means to actually tackle the situation. We have to think in a broader way, not than just dealing with the practical some small interventions. So that's what only I say for the WIA.
Okay, it was a very nice question, but difficult.
Yes.
We won't leave you. We have another by Dr. Ramesh Kumar. Wonderful insight. Uh, Human versus conflict. So what is the ultimate solution?
Sir, means I actually, if you will ask that the ultimate solution, I don't think we have right now any ultimate solution to resolve this conflict. Overall, like my study suggests that it's actually this conflict is getting worse, not in terms of just the humans and the conflict, but with the authorities also who are involving. So what we can do is we can at least build that trust with the community who is living or who is actually affected by this conflict, and try to develop some interventions. Still I will go to that managing the garbage and awareing them that not to feed the rhesus macaques. That I only see as a solution, and it is going to be very long process, not immediate, not even a decade. I believe it will take more than a decade to, if we will go for this solution. And in that period we have to develop some the habitats of rhesus macaques, and means then develop some habitats for rhesus macaques, and some maybe the sanctuaries that we can develop.
Thank you so much for that.
Thank you.
And we are having so many questions.
Okay.
Later.
Thank you, sir.
And, the next question is for Professor Yusuf. Question asked by Dr. Ahmad. Is the concentration of major species to the central area of the PA influenced by the supplementary food provided by the department? Your observation.
Yeah, thank you, sir, for the question. Yes, obviously, as I said, that there are feeding points in the area for macaques. So they keep the food, bananas, and everything on the feeding points. But what happens is that other mammal species such as cattle, spotted deer, wild pig, they come to those places and eat the leftovers left by the macaque. Yeah, it does. The main reason for the concentration is the feeding, and they are feeding not only macaques. So by feeding, they are not only increasing their numbers, they are adding fuel to the fire. So it will explode one day. I believe. Thank you.
Yeah. The next question asked by Dr. Manjari. Do you recommend to clean out the wall? Excellent talk. Good humor.
Thank you, ma'am. That's really nice question, actually. Yes, as you saw in the camera trap, there was just a break in the wall, and you saw we just installed camera trap for a few days, and we were very surprised to see so many number of mammals crossing that area. So I'll not, and obviously I cannot, it's not up to us to break the wall, it's up to, yes, no, I'll not break it. I'll suggest creating small underpasses for the mammals. We have many times seen nilgai jumping over the wall and like getting injured in very bad ways. So I'll not suggest breaking it because it's more of a political stuff. So I'll not go into that, but I'll recommend creating underpasses or maybe leaving places at certain areas where animals can move freely, as we do.
Okay, the next question asked by these garbage dumps in and around the Livelihood wildlife sanctuary, they are known to influence the distribution of food like wildfish and chaos. And the second question is why sambar and striker are only distributed in the southernmost state in public?
Yeah, thank you. The garbage dumps are along the boundary of the area. So what happens is that there are domestic pigs along the border. They come there. So what we have also observed during our field survey, wild pigs come and they have bred with them. So there are breeding happening between wild pigs. So there are no garbage sites within the sanctuary apart from those feeding centers, but there are on the edge, on the lines, there is like they are throwing everything into the sanctuary, which is obviously attracting wild pigs and nilgai, but not other animals because there are a lot of humans and a lot of disturbance occurring there. And for the second part of your question, so as I said in my presentation also, the southern part of it's under the army actually, eco task force. So it's entirely under eco task force, so they don't let humans go into that area. The first reason. The second reason is that there are many mines in that area because Bhatti village, Bhatti mines. So because of that, Bhattis are very deep. So we had a great trouble getting into the mine to install cameras. So local people do not go there. So these are two main reasons why there is a lot of Sambar is there, because there is least disturbance in that area, and there is also good connectivity. Yeah, thank you. Thank you.
The effect of tourism in already degraded aquatic
The use of motorboats can hamper the ecology of river ecosystem and hence influence the biodiversity of aquatic species. But the dolphin safaris that has been adopted in the Jalaj are basically the manual boat safaris, and they does not matlab the boatmen they are well trained to ensure to maintain the ecology of river system. The ranges that they are allowed to travel and the precautions they have to take while taking the visitors on the sites.
Do you think that the higher incidence in Assam is simply due to higher reporting as compared to Chhattisgarh?
Basically, in Assam has the highest number of elephants also on comparing all three states. So that could also depend on the number of elephants present there. And in Chhattisgarh, the population of elephants the population migrate in Chhattisgarh from Jharkhand. So there are no residential population of elephants in Chhattisgarh. They are coming from Jharkhand only. That's why.
The next question asked by Cheetah, the diploma. Your study highlights the positive correlation of forest cover and high it is due to the quality of forest service the elephant. Degraded forest and the availability of other species, water.
As we have shown in the land use land cover map of all three states, we have seen the decrease in forest cover and more numbers in the human encroachment into elephant habitats. It's maybe because it's of that reason. The forests are fragmented now, and elephants migrate in season. Their seasonal migration patterns affect they go from the elephants migrate for their resources, so that's why.
Next question asked by Sayakata from Diploma. Presentation. You have mentioned about the need for site-specific mitigation methods. Various such methods like the solar fencing and teleferencing change in crop changing in property data, the bridge villages, SMS with the alert system, quick response teams for elephant driving, etc. have been tried out and tested in states like Bengal. However, the deaths have not reduced significantly. Any input on the mitigation methods such as
Thank you for the question, sir. I can give you an example of Karbi Anglong district in Assam. They have introduced, for reducing the conflicts, they have introduced the crops which elephants like before the agriculture fields. So the population is coming there, they are eating, and they are going and they are migrating to the path where they are traveling from from the way they are traveling. So in Karbi and in Dibrugarh division, they have installed hanging solar fences, which will not harm the elephants as well. They are also help in those divisions. These two innovative they are working on, and in Assam, everyone is saying कि वही introduction of those crop species before the places of conflict will decrease the conflict in those areas where the maximum conflicts have been happening over years. Yes.
Thank you. That's all for you. Next, Prajapati. The next question asked by Anila Chak from George from the Livelihood.
Thank
Well presented. How do you identify?
Thank you for the question, ma'am. So we identified that Dugong individual based on the body marking that are present, because जो cuts होते हैं in the tail or flipper parts that can't be healed because it's a permanent cut. So based on that, we have identified the individual.
The next question asked by Galliformes. The first question is how these reporting are verified. Second question is what after incorporation can be made from the citizen science collection data.
Thank you for the question, Said. So before involving them in the Dugong monitoring program, we sensitize them and we train them see how the Dugong look like from the boat-based survey, whether it fishers community who are sightings from the boat-based or underwater sightings that is only be done by the scuba divers, and for the aerial sightings. So we have trained them to how to identify Dugong. So later, then we have involved in the Dugong monitoring program. So this is how we got the verified report because they are trained to identify Dugong. And the second.
By using how did the Dugong affect population? Second question, what was the reason of individual not given?
Thank you, ma'am, for the question. So we have when we did our interview survey with the fishers community. So before that, they were saying there was a Dugong before tsunami. There was a social structure because Dugong वो time पे herds में दिखते थे. But after tsunami, the seagrass meadows also got destroyed. So maybe this could be the reason because of their population got affected. Because the food they eat, the only primary food they eat is seagrass only. So अगर जब seagrass ही नहीं रहेगा तो this might be affect the population of Dugong. And the second question, the injury mark. So this could be, might be from any region. Maybe bites from the sharks or from the propeller cuts. It can be any cuts. So that's how that mark is there on the Dugong body.
That's all. We thank all the speakers for their wonderful presentation. And, before we move on, basically, we will ask the question. This is a question as a suggestion to Kalpana. It's like to ask you how to interpret the findings of livelihood density of forest in the case with the high mortality due to or roads near forest increase the interface.
clinical activities, we may need a focused analysis to examine. So basically, to kind of start off an excellent presentation, maybe a Kaziranga and now request Dr. Cheetah.
It was a wonderful session. Watching and listening to all those presented narrations of higher conservation was a rare great experience, particularly in this time period where humans are everywhere and we need to observe the remaining biodiversity that we have on this planet. I commend all the presenters for their excellent work and I wish you all the best for the future.
So I request to proceed the next two slides. Yes, we have reached almost the end of the last 8 technical section on human dimension in wildlife conservation, which is a very, very important topic in the wildlife science. So far, we had observed diverse ecological and conservation challenges in about 6 various topics presented here, which also include terrestrial as well as the aquatic habitat, which include freshwater as well as marine landscape areas. Each of the presentation offering unique insight into wildlife interaction and conservation challenges. Starting with the Kaziranga Tiger Reserve, we have explored livelihood strategies, balance conservation needs, while addressing vulnerability faced by fringe communities. It was an interesting presentation followed by in Solapati Wildlife Sanctuary. We have had 2 presentations. The first one was discussed about the human-macaque conflict presentation, a case of complex interaction that human with a target mitigation and the community engagement, which is required to solve the issues. And followed by we also examined the mammalian diversity in this urban sanctuary, setting light on species adapted to the human. And both the presentation was focusing on the aquatic biodiversity in the Ganga region, where integrating livelihood and conservation effort is the key for success. And fifth presentation was covered related to the human-human conflict in Assam, Chhattisgarh, Jharkhand, which was highlighted the tragic consequences of habitat encroachment, which was leading to the human-elephant conflict. Was very much sensitive issues in these days that was excellently presented. Finally, from Peninsular India to the islands, we are come to discuss about conservation in Andaman and Nicobar Islands, which underscores the potential of citizen science to manage the mammalian species of the remote island scenario. So all these 6 topics were very well covered under the human dimension in the wildlife conflict scenario. And I congratulate all the presenters for their wonderful presentation and all the supervisors for conducting this study. Once again, best wishes to all the presenters. Thank you. Thank you very much. First of all, I want to compliment all of this excellent presentation. I have 3 points, 2 to 3 paragraphs. You can take it as a comment or suggestion. So one is that I think we conducted presentations, but for the last session, Dr. Leonard, now Dr. Shivani, this is, I think, in all main channels. Thanks for having that diversity to this. I think it's important to keep diversity, equity in mind. But then you compensate with the kind of presentations I was looking at it. I think more than 60% of the presentation made by human researchers, which actually says a lot about this institution. And the second point I want to highlight is we talk a lot about the presentation getting into visuals having food, but I think it's one more point about institutes innately common. I can tell you, when I was a researcher, I was both here in Wildlife Institute and made my first presentation. I see now interns making a presentation. And people within 3 months they're making presentation. I think that's the kind of innate environment the institute is actually talking now. There are 3 days. I really compliment because I never thought they should be able to do this because they are gone for me. But this kind of 3 days anniversary and making it now, I think that's up to you. I'm really happy to have the institute. About precise and short messages presentations. I think usually when we have presentations, there should be a take-home message in short. But often I look at myself. Whenever it comes to some sharing. It takes 2 lines. But most of them I don't understand. But this time was very particular. Because he's sitting here and writing the diary, he talks about horse. Horse made up based on 1971. I think that the diary is there. We will be saying about the diary. I think that's an excellent thing. And what I want to say is, when we communicate, I think sometimes it's very important for us to be precise. And today, when I went to teach them, Dr. Parag challenged me to Tamil. So I really need to. Basically, if I say something, it's actually, if I blame it on it, that's what I want to say. I want to take an anecdote which goes from Southern literature back in 2004. It was a very famous, enlightened lady, she's called Human. And a lot of things, a lot of small points, very powerful. In schools, we all read and listen. So one of the occasions, it happened that she was supposed to kind of price the particular thing. I didn't know anything. So when she said, she actually uttered 2 words. That was Varapuya. Varapuya is a fun. Puya is the race. She was supposed to bless her, say something, but she said just 2 times. Then it didn't make any sense to you, but she continued on. Varapuya, Nehware, I will translate later because I need to have a flow. Varapuya. So Varapuya, Nehware, 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 Kudivare, Kodvare, Now we have, so now you listen to our director in the independent state, and also yesterday, he took a lot of measures, welfare measures, starting from the fundraise and resources grant and so on. And something else I'm saying, if I teach somebody says, brains is the only one. If somebody says, it has something, what exactly means, she was trying to say, then the one raises the one. It's a kind of analogous to the productivity of water. So, Varapuya means the raise of the water. Okay, in our study, I think what we're trying to do as a researcher, I think the kind of social research that we have, and the diverse research. So, the one, when we talk about one, we talk about multiple things as an institution. There is the PH lab raising the bar, there is the MBA lab raising the bar, there is the University lab, there are Samrat's labs, there are labless labs. So many of us are working together. Okay. So basically the idea is there are a lot of research is going on. I think the cross-sectional research is happening. Only probably many of the faculty members don't even talk to each other, but the students actually have an opportunity to talk to. And this is an opportunity. I think I pay so much emphasis on IAS because this actually provides much more opportunity for students. So that's my story. So basically, when so research happens, when she talks about a productivity, yield of the paddy, that's what she says. When the paddy grows. When basically the people are happy. Okay. When people are happy, the governance is respected. When governance is respected as an institution, it glorifies. So that's an excellent narrative that Thank you so much.
Thank you, sir, and we thank the research coordinator, the director, for this opportunity. Thank you. Thank you, Dr. Ramesh. Both Dr. Ramesh. So we are just starting to begin the competition session. I request Dr. G.S. Chair to take the desk and I will give the company session.
as the custom goes in this auditorium and institute, uh, we invite few people to give their comments on the. So to begin with, I request I've been attending almost all our gatherings, and it has been a wonderful 2.5 days discussing 8 thematic topics and 48 presentations. And one of the things that has really increased my knowledge because one of the things today's era is a multidimensional. And in the past, in the IRS, our whole used to be almost 50%, 30%, but this time, I said almost 20%. So people have realized it's not my work, but other works can decide to be a much more better understanding of the science. And it might be possible to integrate during your present. So this is one of the things I should cover with everyone to be listening all the talks, and this is a wonderful thing with communicating to everyone. Another thing in the past, we used to have most of the talks about the carnivore, or even the researcher used to come back. They used to say, I want to come tiger, anything like that. But this time, if I see, there are only 20% presentation on carnivore. The rest of the presentation is ranging from new families and ranging from invertebrate community. So this is the one thing is one should understand that this era is multidimensional and to try to invite as much as the knowledge of the different discipline to enhance your understanding better. Another thing, what the new eras on which you have seen the new tools are coming day by day, and it's nice to hear one of the tools like what Samrat Group has talked about the hormonal. Okay, so this is a new thing which if you are working in the field might be possible to. You may not get exposure, but you may collaborate with someone who use the hormonal ecology to understand reproductive biology of the free living animal. So these are the new things which have come, and another thing, the last of what you heard is the citizen science. This is another way which you can enrich your data collection. So basically, the new thing which I heard one of the urban ecology is a very good thing. Another thing, the urban ecology, that's one thing. Community participation because it enhances the overall resources habitat, which might change your use of the animals or whatever you are looking. And the third thing, what I got me interesting thing to hear, because this is the first time I heard the use of overpasses by animals. So this is the first study which came in India, where we used to have some kind of the, worry whether the overpass will be used or not by the wild animals, and we have invested a lot of money, and we could see the interesting findings the wildlife overpasses are used compared to the other passes. So this is another new thing which has come in our country, and it enhances our capability of confidence. One of the important things what I realized that most of the researchers probably considering the first time. And amount of the confidence I could see is it was not very large. This is the one thing what Ramesh has said that they probably allergic environment, interaction with other researchers, and that could be one of the reasons that I could see the confidence among the researchers. And I remember whenever in the past we used to say researchers, please see that you have used the real font size which are visible from the back of the sea. But this time, every presentation was a perfect font size. Even in the last row, you ask them, they could see the font. So this is one of the things you have got a cultured presentation, and it made the, I don't know whether you have done the rehearsal or not, but I don't think anybody has done the rehearsal, but you have tried to incorporate all those things, things which are very, very important. One of the important things which, what Thomas has said yesterday, in the past, we used to have a very simple statistic, either the ANOVA, maybe PCA, maybe chi-square, like that. But this time we have got a more predictive model. It's a very, very good tool. It allows you to make understand the positive factors, but at the same time, what the study Dr. Kamal has said, we have to integrate in our influences the ecology of the species. So it is not like that only we are skewed toward the only predictive modeling and try to see whether those predictive modeling or positive factors are really making a meaningful contribution to the consumption or understanding the specific ecology of Earth. So these are the things that tools are very, very powerful. It allows you to make more better inferences, but try to do not forget the ecology of the species and which varies species and category. So these are the few suggestions which I would like to make. And one of the things I would suggest is
especially y-axis results are not there. So be careful that you convey the message what those y-axis data is there, because if you have got the 0, 10, 20, 30, 40, but you don't know what is there. So this is not in all, but there are few projects with accuracy, and this is the one thing, be a little bit careful and try to see whether all results are there or not. And lastly, I would congratulate to everyone having the wonderful presentation, the confidence. This is the really, and keep this confidence up in your life. I think you can win the game and we can really have a better presentation in the future. So wish you all the best to everyone.
Now request my colleague Dr. Parag Nigam to speak a few words. And I'll tell you why I picked up Parag Nigam, not other colleagues. Dr. Nigam was happy to be seen in this class. So I got curious and went to his room. He was very careful watching the institute website presentation. So Dr. Nigam.
So good afternoon, it's wonderful. 2.5 days have gone very fast, but I have been talking between offline and online. But having seen the presentations, I can compliment each and every one of you for presenting very well, and both from WI as well as SECOM. All the researchers that you work hard. A lot of analysis, a lot of strong analysis. Something that people just skip through, but then definitely the conclusions were still, we could really take in the message going in. The qualities are really good. Dr. Boyd really spoken very well about the confidence, the way it was presented, everything. So I was seeing the comments on the online coming in, people really appreciating those who joined online, and that was a very positive for all of us. But having said that, what comments are also saying the same thing. More sweat in the field, more field-based data, and a little bit of invasive things have to start with. A lot of non-invasives are very good, very nice. People start with the endocrinology to doing observations. That's really important for ecology. But I would expect we have been doing for the last 5 years a lot of invasive ones where a lot of data and other things were presented for. So definitely encourage all the researchers to really push it back to component also, and definitely addressing those wildlife health issues. So overall, wonderful, and looking forward to getting next year for the ARS that will get in. A lot of good wishes to you and the PIs will be presented. And we're excited about all this. Thank you so much.
We had three guests who really agreed to be with us entirely. So I will ask one by one, three of them to give their comments. You can just turn around and look at them.
It was a very interesting experience for me. Actually, when Dr. Vimas came for IAR, we had a lot of preliminary studies, exploration, but really surprised by all congratulations to you. And of course, your peer mentors also doing it. Every one of you time over this. I wish you all the best. In the national or international forum. Regarding the diversity of studies, very impressed. As Dr. Goyal have commented, earlier there used to be species-centric biology, cardiology, now it's hard to see the integration with the discipline which is probably very hard to augurs well for the future. You brought
when cognitive states are working actually become factors of responsibility. That will make factors. Thank you.
Thank you, Dr. Ram. May I please invite Dr. Lucy?
As we go on it, extend my sincere every visit to Dr. feels like there's always been uh, the market what is 2.5 days we have seen commented research that has been presented ranging from Himalayas, the Himalayas, to coastal region, islands, and focusing on the important and unique species. Every research has done it in talk. We have presented presentations by and reflect hard work and dedication every year. I compliment each of you for the wonderful presentation, and I wish you all continued success in your future. We have tried to assist each one of you with fairness and open mind, and I compliment all of you for the good work you all have done. So once again, I thank WIR for this wonderful opportunity, and I look forward to seeing the more focused and contribution.
I now invite Dr. Cheetah.
Hello, and thank you for having me here. Thank you, Vash Chandra, Director at WII. I'm from ISL Mohali, and the best that I hear in ecology and evolution is Drosophila, which is also fine. So this is, you can imagine how enriching this is for me. It's the nutritional stress, intellectual, has been satisfied here. So I go back very happy. The work that I saw here takes me back to my own PhD days 20 years ago. Very grounded fieldwork, a lot of very cautious analysis, and I also like the fact that everyone was very receptive to suggestions and constructive criticisms that were given. Another thing that I see sometimes it's lacking when we are younger and we want to prove ourselves. So this was also very fascinating for me. I have learned a lot, so thank you very much, all of you. Can you also please teach me how to make these very pretty slides? Please come to Exxon teach us, teach the PI how to make these slides. They are so fantastic, such pretty slides, not just because you had the title and the beautiful words, but they were organized very nicely. The themes were coming out very nicely. And the maps, oh my God. So I don't think we can do it. So please teach us. We want to learn. So thank you very much. Everybody.
Thank you, Dr. Manjari. So I request Dr. Vare.
Okay, thank you very much for the advice. Firstly, I would like to profusely thank the WIA for giving me the honor of chairing that seminar. Secondly, I also congratulate Director WIA, team, registrar, and entire team, including the coordinator, for putting a wonderful seminar here. And it's really happy to see the progressive advancement and development in both infrastructure, physical, as well as EV system, as well as the coordination and the time and everything, and all the wonderful presentations. It's really good feeling for me because I have also attended all the internal research seminars. In fact, this internal research seminar has also improved in quality so much that it will be very difficult to differentiate in terms of quality between the internal research seminar and the main seminar. The researchers who are going to make presentation in the main annual research seminar will really find it difficult to cross the bar as been said by the students in this presentation. Despite a very, very short period of study, they have done a very, very balanced work of field work and data collection, data and documentation, as spoken by previous speakers, that the quality was really, really nice. I must compliment them. And it was really, for me, also really, learning experience, learning about Malaysia to Andaman and Nicobar Islands, from Jaisalmer to Arunachal Pradesh, then of the nation. As also mentioned by Dr. Goel, that 10, 15 years ago, the students standing here had less confidence, especially in terms of the quantitative tools and statistics and the analysis which they had performed. Earlier times, whenever there was a question on statistics or use of particular tool, the students would look towards Professor Kamar Kureshi or other colleagues to explain on their behalf that how they did it. But this time, most of the students knew what they had done, why they had done, and the reasons for selecting them. I hope we will continue this spirit and also remember Professor Kamar Kureshi's comment that don't use the particular tool just for the sake of using it, but you will see the logic and also analyze what are the limitations and improve further. Lastly, I would like to say that all the PIs and students have really done very, very hard work, and I'm sure you will collect all the questions which could not be read yet. You will collect them and do further analysis and do your presentation, find the appropriate answers for all the questions you could not get there or you could not answer properly. And I again wish you to all the best and keep this passion, this interest and this enthusiasm for your research, for conservation of our certain environment, our habitat ecosystems. Wish you all the very best, and I thank once again for this opportunity and the team. Thank you very much.
Thank you so much, sir. Now, the last item of this seminar, vote of thanks. So I'll go there and extend my vote of thanks to all of you.
So the reason of coming here and extending vote of thanks, बैठे बैठे thanks नहीं बोला जाता है. अगर किसी को धन्यवाद देना है, खड़े होके हाथ जोड़ के और बड़े विनम्र भाव से देना पड़ता है. So that's why I extend my sincere thanks to the following. At the outset, let me thank Santosh, my office assistant, who has been roaming around last 2.5 days, collecting the slips, giving it to him. I extend sincere thanks to Rajendra Kumar, Pritam, Guljar, Sekhar, Hijab, and Sachin, who are in charge of this auditorium. There are two minor glitches. Light electricity went off twice, but it is not their fault. Fault belongs to the power corporation, but everything was managed so smoothly. I have sat in this auditorium, AC was not working, audio system was not working, all those things. I was very happy that everything went in very precise manner. Thank you very much. I thank Sri Vinod Verma and Sri Gaurav of AV unit. विनोद वर्मा जी को आपने देखे नहीं होंगे, बहुत कम टाइम आए, ऊपर बैठे हैं. And he has been controlling the entire audio system in this, and Gaurav has been taking some beautiful photographs. I will email these photographs today in the afternoon or evening to all of you. So thank you very much. I thank Dr. Pannalal, Sri Dinesh Singh Pundir, Sri Harendra, Sri Ankit, and their team sitting in the front row here. Not only making this new system very, very effective, how many people have attended? Can you just tell me briefly online? Around 2,000. Around 3,000 people have viewed our annual research seminar online. And thanks to our IT support team for making this possible. I truly appreciate. Or believe me, Monday morning when I came here, I had no clue ये कैसे होगा, क्या होगा, कैसे करेंगे. I came little early, but before I came here, everything was set, everything was ready. So that was a big relief. Otherwise, I was very tense on Monday morning, क्योंकि Sunday रात तक मैं इधर उधर भाग रहा था. But then you made our life, all our life, very easy. Thank you so much. I thank Srimati Alka Agrawal and Sri Ganesh Chibar of the academic cell for assisting me with this seminar. Thanks are due to Sri Virendra Sharma and Sri Narendra Singh Bist. They prepared the first draft of this program and abstract document that I have circulated. And then Ms. Swasti Joshi, who has been working with Dr. Vishnupriya, she gave the Midas touch to this entire program and abstract document. So thanks to all of you. I thank all the MSc students. We have 3 batches of MSc students, big strength, nearly 60 students. So मेरा केवल शिकायत है 19th MSc student, 19th batch, only 12 of you have registered. So I presume that only 12 of you have attended. So please make it a point to attend these seminars. And when you are present physically in the campus, please come to this auditorium and attend this. Do not do the mistake of sitting in your room or chilling out in your room. If you look, this auditorium is a much better place to chill out than chilling out in the room. Okay. Thanks all the students for helping us out. I thank all my faculty colleagues for their active participation in this seminar, making this seminar very alive through asking questions. I thank in particular Dr. Vishnupriya Kolipakam, my associate research coordinator. She is not present here, but she has been helping me all through. If I am able to conduct this research seminar, it's primarily due to her help. I thank the members of the committees that are set up for IIRS and ARS, particularly the program committee, the abstracts committee, abstracts and review committee, and the catering committee. Regarding catering committee, normally we look for chicken in the biryani, but yesterday we were looking for rice in the chicken biryani. Okay, thanks to the members of the catering committee. And if you are fed up with chicken in the biryani, आज आपके लिए बकरे ने जान दिया है. So after this, please go back and do proper justice to the supreme sacrifice that has been made by the goat. Okay. I thank all the session chairs, the co-chairs, and the facilitators for conducting this seminar in the most effective manner. And I'm very proud of my colleagues that they have managed to finish this seminar dot on time. Starting from the first session to the last session, we finished at 1 o'clock. We started the concluding session at 1 o'clock. Thank you very much. Thanks to Dr. Raman Kumar, Dr. Manjari Jain, and Dr. Khurshid Ahmed for agreeing to come with us. I hope we have taken care of you well. It was a pleasure to have three of you with us, and we look forward to your participation with our or working with you in future. My sincere thanks to Dr. G.S. Rawat, who has been my guru. When I joined Wildlife Institute in 1991, Dr. Rawat was our course director. And very long association, more than 3 decades of association. Just one phone call, Dr. Rawat told, let me check, I have another engagement. If it is happening or not, I'll get back to you. Within 15 minutes, Dr. Rawat confirmed that I'll be here. It's a pleasure to have you, sir. It's an honor to have you with us. I thank Dr. Satyakumar, S. Satyakumar, Scientist G and Registrar of this institute for all the administrative support. It was very smoothly conducted, absolutely no issue. Just one WhatsApp message, not even phone call, things were done. Thank you, sir. I thank our Dean Madam, Dr. Ruchi Badola, for her support all through this program and all through this research activities that are going on. And a big hand to our director, Sri Virendra Tiwari, for his outstanding leadership and motivating all of us to deliver our best. I derive tremendous inspiration from your leadership quality, and I'm sure all of you are doing the same. I request everyone present here to give a standing ovation to our director. Please. It was a pleasure to, it is a pleasure, not it was a pleasure, it is a pleasure to work with you, sir, and I look forward to the forthcoming ARS under your leadership. And finally, my heartfelt thanks to all the research scholars. It has been your show, and you have put up a fantastic show. Thank you all. अभी एक और announcement, दो और announcement है. Deadline for ARS abstract submission is on 15th of September. So please submit your abstract well on time. Don't wait for 12 o'clock in the night. And then let us know कि सर ये काम नहीं कर रहा है, वो काम नहीं कर रहा है. So please start submitting from now onwards if you have not done. So 15th is the deadline. And the last announcement is if you don't mind, if you can come back to this auditorium, say around 3:30 or 4 o'clock, whatever is convenient to you. I would like to have a informal feedback session with you and interaction with you so that we can work on further improvements in the ARS and make things better. Thank you for last 2.5 days, very patiently sitting and
So big thanks to Dr. Vivan Pandey.